break. It's actually just 2 o'clock. I'll give you guys about a minute more to settle, and then we can get back to it, right? I hope you guys enjoy the break. Close my door. And to allow a couple more people to get in. Yeah, but how is it so far, guys? What are you guys thinking so far? Is everybody asleep? All right, so I'm gonna send a message in the group to everyone that we're back. All right, so we can get started, right? So all right, can someone please read the question?
Using annotated diagrams, compare the structure of an unspecialized plant cell with that of an unspecialized animal cell.
All right, so we're gonna start drawing our cells. So if I was to draw a plant cell, how would I draw it? How would I start? I want to start drawing a plant cell.
The outline of it would be a square.
Not necessarily a square, but you have a very geometric outline, right?
Would it be like a rectangle?
Yeah, it's very geometric. It's not perfectly rectangular, but it's very geometric in shape. So that's how we'd start. And then what else would I draw?
So draw another one similar. Membrane.
We have to complete this outer part first.
Complete.
Right. Say if I was to label this, what would I label it as?
Cell wall. Oh.
Cell wall.
And then cell membrane.
Cell membrane. All right. So what is in my plant cell?
The big vacuole, the large vacuole.
Chloroplast, the nucleus.
Wait, give me a second, please. Large central vacuole.
Cytoplasm.
All right. So we have cytoplasm.
It needs to be a nucleus.
Chloroplast, nucleus.
We have a little chloroplast with a little thing like that. Chloroplast. How would you describe our nucleus?
Like that.
How would you describe our nucleus?
Has genetic.
As in, how would you describe it versus an animal cell? It's a peripheral nucleus. We have a peripheral nucleus. What do we have around the nucleus? Peripheral? All right, so you know what the word periphery means? So when something is on the periphery, it's on like the outskirts. It's not in the center. So central versus peripheral. So it's kind of like close to the wall versus it's in the center. Cool? It's off to the side. All it means is that the nucleus is kind of off to the side. All right, so any more organelles?
Starch granules.
We have the vacuole.
Starch granules.
Yes, perfect. We have the starch granules. But that's not an organelle. Anything else?
Yeah.
What's it called?
Mitochondria.
Mitochondria. What else?
Ribosomes.
Yeah, we have the ribosomes. Yeah, give me some more. I'm not gonna draw the ribosomes yet.
I think that's
What surrounds the nucleus? What's kind of on the outskirts of the nucleus?
The nucleus wall.
You guys have never seen something that looks kind of like this on the outside of the nucleus, kind of like surrounding it. The rough endoplasmic reticulum. Or sometimes they call it the RER. You've never seen that?
Yes, sir.
And what's under is what? No, not necessarily, but it's one of the organelles. So I want you guys to know it. Just know. I don't want you guys to just say like you've never like you go and that's the first time you're seeing it. You also have a smooth endoplasmic reticulum. What's the difference between the smooth and the rough endoplasmic reticulum? What is on the surface of the rough endoplasmic reticulum? Do you guys know?
Sure, I don't think we do that at this level.
Oh?
I don't think we have that at this level.
Oh, all right. What makes it rough is ribosomes. You have ribosomes on the surface. So, and you also have in the cell, you have free ribosomes. So you have ribosomes. You have ribosomes. What else? You also have Golgi. You have Golgi apparatus. So those are pretty much Golgi apparatus. So if we're describing a plant cell, what else? You also have to have chloroplast. Chloroplast, I have starch granules, cell membrane, Golgi. Yeah. So if you were to draw this, bam, excellent. All your marks. So you guys are saying that you don't really have to know about the RER and the SER and the different organelles?
Sir, I don't think so.
Okay, all right. But I mean, it can't hurt to know. It's just, you know, another thing. If we were to draw an animal cell, how would you draw it?
Sir, it has an irregular shape, so like circular and something.
Like a sphere.
Yeah, just irregular. Perfect. Irregular. Cell wall?
No, sir.
What's the cell wall of the plant made of, by the way?
Cellulose.
Cellulose.
Yeah, perfect. Cellulose. So cell wall for the animal cell?
No, sir.
No? Are you guys sure?
Yes, sir.
No cell wall, just the cell membrane.
Guys, just a cell wall. I'm kidding. That was a test. Nobody's laughing. Okay. Cell membrane. There's no cell wall. So the cell membrane, how would you describe the nucleus? It's a central nucleus. In the plant, you have a peripheral. In the animal, you have a central. Plant, P, peripheral, P, nucleus. Cool. As I said, guys, you can find ways to remember the stuff. See a nucleus, around the nucleus. I'm just gonna put in the organelles this time. You have your your rough endoplasmic reticulum, your RER, you have your SER, you have your Golgi apparatus, which looks something like this, and there's always something to vesicles body off it. This is your Golgi. Give me some more organelles, please, guys.
Small
Vacuoles.
vacuoles,
Small vacuoles. What's in a vacuole? Anybody know?
cell
The waste material. Or probably like it stores stuff for later use.
Stores water, cells up.
In an
Yes.
animal cell. In an animal cell. What's in a vacuole? Anybody know?
No, sorry.
You have enzymes in there. Just enzymes. It's not anything big or anything like that. I was just checking if you guys know. What?
So say for the plant, it is the cell sap and water.
Yeah. Cell sap. That's what's in this vacuole. Cell sap. The major constituent of cell sap is water. So we just call it water. So a vacuole, what else we have? Little mitochondria in there. We have
Granules.
what? What granules?
Is it glycogen? I don't remember what it's called.
Glycogen granules. Perfect.
Yeah.
Was it? Oh no, you guys wouldn't know. All right. Glycogen granules. So you have your you have your little ribosomes. I'm about to ask you guys what the function of all these stuff is, by the way. So think about it. What else? Do you have anything else?
No, sir.
Yeah, I can't think of anything else either. So I didn't label the cytoplasm. All right, so let's start from the top. What's the function of the cell wall?
To give the plant cell a fixed shape
Sure.
so
Yeah,
that it
perfect.
doesn't burst and stuff like that.
No, you can just leave it at the first part to maintain the shape. Sure, that's fine. Purpose of the cell membrane.
To protect the contents within the cell.
Anybody
Or
else
to-
have an idea?
Allow substance in and out of a cell.
Yeah, to create a barrier between the cell and the external environment. So protection, yeah, but I mean it's a barrier. Like if you put something in a plastic bag, it's not gonna come out and nothing can go in. Same concept. What's the purpose of the vacuole?
For support.
What's the actual purpose of the vacuole in the plant cell? It just stores water, guys. It's just, you keep it simple. Kind of keep it simple. It stores water. By the fact that it stores water, it's able to support the cell. But its purpose is to store water. What's the function of the chloroplast?
So it contains chlorophyll.
For photosynthesis.
Yes.
Yeah, it contains chlorophyll, but it's also the site of it's also where photosynthesis occurs. So that's good as well. What's the function of the nucleus?
Control the cell activities.
Sure, yeah, that's good. Function. You guys don't know the function of the RER, do you? It's the same as the function of the ribosome.
Sir, I don't know the function of the ribosome.
Anybody know?
It synthesizes protein, something like that.
Protein synthesis. So the RER does the same thing. The RER is just a bunch of interconnected ribosomes. So the RER and the ribosomes have the same function. So yeah, the ribosome just synthesizes proteins. Golgi, what does that do? I'm gonna skip the SCR because you say you haven't done the endoplasmic system. So what's the function of a Golgi?
So we don't do that.
You don't do function of the organelles at all?
So
That's
not
not.
the guy.
I saw cell specialization on the syllabus. All right, I'll just tell you. Modification and packaging of proteins and lipids. If they ask you that question, that's the answer that they're looking for. What's the function of the cytoplasm?
All the organelles.
Ideally, like substances, the organelles.
Sure, but what's the actual function? Yeah, it holds the organelle. All right, give me another function, a better function. Doesn't it help to maintain the cell shape? Stuff like that. Yeah, right, guys. Also a lot of cell reactions take place in the cytoplasm, right? But that's just something to know. My gosh, what just happened? Also a lot of cell you guys can still hear me, right?
Yes, sir.
I agree. So a lot of cell reactions also take place in the cytoplasm. So that's also important to know, right? So with respect to the mitochondria, what's the function of the mitochondria?
Provide the cell with energy.
Yeah, perfect. All right, so yeah, we can move on from it. So yeah, we have a really simple diagram here. So look at this kind of diagram for like a bit, and then I want you to answer these two questions to follow. You guys have 10 seconds.
One is osmosis.
Okay, looks like you didn't need 10 seconds. Let's explain that.
Go back to the question.
Wait, anybody can answer?
Yeah, anybody can answer.
Wait, what?
Explain why you say it's osmosis.
Right, because in plant cells, when they are placed in a hypotonic solution, water moves into the plant cell, causing the plant cell to become turgid.
Cool. But no. You're kind of mixing up your terms. But I get where your head is at. So good.
Oh wait, wait. Is it hypertonic
what did you
solution?
say? Hypo or hyper? What did you say?
Hypo. HYP.
Okay. Oh, yeah, I didn't hear. All right. So yeah, you're correct. So yeah, perfect. Yeah, but you never have to go into all of that. You could have just said, because I mean you're placing the cell in a solution and the cell getting bigger, and that's the whole process of what osmosis is. But yeah, but I'm glad that you were able to follow up with that. For number 2, can somebody give me a reason for that? Give me 1 second, guys. You guys can still hear me, right?
Yes, sir.
Sure.
Cool.
Osmosis is important because the plants need water in photosynthesis.
Sure.
Keeps it.
Yeah, of course. Yeah. Allows. So all right, put that in better words for me, please. Can you help me out? Like put it in a bit better terminology. Use up your biology terminology. Anyone?
Wait, what number are we on?
2.
Oh wait, but I did that one already.
So explain why osmosis. All right, what's the answer?
So like when the plant cell is placed in the hypotonic solution and water moves into it, it becomes turgid.
On part two. So explain why osmosis is important to plant cells.
Sure. Is it because it allows for water to go into the cell in order for photosynthesis to take place? And allows for excess water from respiration to leave the cell, like the wastewater.
I like where your head is at, but I wouldn't put that. But I do like how you're thinking. But no, don't put that. Don't put the second answer. I'm not gonna get into the. All right. You use water. You use water for other stuff. You don't really want it leaving the cell just like that. You don't really want to lose water so much. So you don't waste. You don't really have much waste water per se. So water. Anytime you have water, you want to use it. So give me another reason, please. You can give me another reason. Joel, use it for
respiration.
Sir, it allows us the plant to remain rigid, turgid.
Yeah. Allows for the maintenance of turgidity. Yeah, I like how you guys are thinking about respiration, but we'll get into respiration in a bit. Respiration doesn't really it's I'll explain a bit. Allows for the maintenance of turgidity. When water enters the cell, vacuole expands, presses against the side, and turgor pressure is increased, so the plant is turgid, so the stem is now upright and not wilted. You guys have seen a wilted plant, right? Anybody hasn't seen a wilted plant, say something, please. All right, say yes if you have seen one.
Yes, sir.
All right.
Yes, sir.
So when you look at a wilted plant, the leaves are kind of drooping. The leaves are drooping, the stem is down low, things such as those. That's not the most effective setup for photosynthesis, right? Because we don't want our leaves to be standing up and upright and broad and at our angles and everything like that. And if it's drooped, the leaves aren't at the highest possible point that they can be to allow for the maximum entry of sunlight and things such as these, and they're also shriveled, which reduces the surface area, things such as these. So it's just stuff that I'm thinking about because I know how a wilted plant looks, and I can see, oh no, that's not what you would want for photosynthesis, right? So things such as those. But yeah, just thinking out loud. So let's move on quickly. Cell wall, plant cell, animal cell. Let's just move to this quickly. Plant cell. Present or absent? Animal cell.
Absent.
What makes up the cell wall in a plant cell?
Cellulose.
Chitin.
Pardon me?
Chitin.
Chitin?
That's for fungus or something. I don't remember.
Oh, chitin. Yeah, you're talking about chitin. No. Chitin is for chitin I think is for no, chitin is the shells, isn't it? The shells of I think it's for a shell. I'm not sure though. Or maybe it is for the fungi. I can just check it out, but yeah, no, it's not chitin. Yeah, cellulose. Yeah, just keep it typical. Yeah. Yeah, nuclei. Describe the plant nuclei. How is it placed?
Peripherally.
Animal cell.
Central.
Essentially. Mitochondria. Plant cell have, do plant cells have mitochondria?
Yes.
Yes, sir.
Animal.
Yes, sir.
Yes.
Chloroplast. Do plant cells have chloroplast?
Yes, sir.
Yes, sir.
Do animal cells have chloroplast?
No, sir.
No.
Okay. State one function of each of the mitochondria.
So before we move on, for the nuclei, that's central, right?
Yes. Yeah, I need to just write better.
Kind
Central.
of weird.
So what's the
Yes,
word
central.
under the plants, under present for plants in the nuclei?
Peripheral. Plants have a peripheral nucleus. State one function. What's the function of the mitochondria?
Provide inspiration.
Provide energy for the cell.
Yeah, don't just say site of respiration. It's a site of aerobic respiration. You guys know the different types of respiration, right?
Yes, sir.
Yes,
Aerobic, anaerobic, blah, 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 blah. Good? Yeah, well, the mitochondria is a site of aerobic respiration. So if they ask for a function of the cytoplasm as well, the cytoplasm is a site of glycolysis. If you want to put that.
No, sir.
I don't want to tell you guys anything. I don't want to tell you guys things that you don't, that might kind of confuse you, but it's just stuff that you can just, remember and say, oh, in the exam, what's one something like that? Oh, I can just put that because, that's what I was told. And you can't get that wrong. So mitochondria is the site of aerobic respiration. Okay. Chloroplast.
Site of photosynthesis.
Photosynthesis.
Yep. Site of photosynthesis. You see, a lot of people would put contains chlorophyll, but how is that a function? It just contains chlorophyll is a description. Site of photosynthesis is a function. It's a photosynthetic organ, organelle, right? A lot of people would put for mitochondria, powerhouse of the cell, but what does that mean, right? So you guys are going to be ahead of the game, right? Well, I hope so. All right, so we can just breeze through this really quickly. Plant cells versus animal cells. These are some of the things that we're going to cover. So looking here, all right, this is kind of a pretty detailed diagram, but we can just see here for the animal cell, you see the ribosome, the lysosome. Lysosome is just, call it a vacuole. Yeah, call a lysosome a vacuole, okay? And there is endoplasmic reticulum, if they ask about that. The cell membrane, we can see it right there. The mitochondria, the Golgi right here. Cytoplasm, nucleus, nuclear envelope. Forget about this word. Forget about this word. Just focus on nucleus. Cool?
Excuse me, sir.
Was someone saying something?
Yes, sir. I can remember the ribosome function, please.
Ribosome.
Protein synthesis.
Yeah, protein synthesis.
Thank you, sir.
Yeah. So in the plant cell, we'll go back and zoom out. This. Yeah. All right, that good? All right, definitely. So plant cell, again, we have a vacuole. We have a large central vacuole. We have our chloroplast here. We have the Golgi, endoplasmic reticulum, the nucleus. Forget about this word. Forget about this word. We have the ribosome. Forget about this word. And we have the cytoplasm. Of course, what's missing here? What's missing here? Starch granules. What's missing here? Glycogen granules. Cell wall. What makes up the cell wall?
Cellulose.
Cellulose. Function of the mitochondria.
Site for
Site
aerobic
of aerobic
respiration.
respiration.
Yeah, cool. Nice.
Can you go
So,
back for me?
all right, these are just the functions. So if you want to take a picture of it, you can. Of this.
Yes, sir. Done.
All right, cool. So yeah, if you want to take a picture of it, you can. Just read it. Again, forget about this. Forget about this. Forget about these things. Psychoplasm. The rest of these are fine. So yeah. Perfect. Yeah, so you can take a picture of this. I mean, it's not gonna kill you to know the function of all of them, but yeah.
So you can zoom in.
Pardon me?
Zoom in, please.
You guys can read it. You guys can see it, right? All right, perfect. So we can just move on from that because that's just go backward.
The next slide.
Which the previous one?
No, sir, the next slide. So the side. Yes, sir.
This?
Yes,
This is
sir.
what you want to see?
Yes,
all right.
sir.
I said these are stuff you can get online. So you just look up a picture, random picture. But just know how to label it and know how to draw it. So just stuff like that, yeah. So, all right, so let's move on quickly. So the differences, I'm not gonna ask. So plant cells have chloroplast, animal cells do not. Cell wall made of cellulose do not have a cell wall. Large central vacuole, remember, small temporary, and let's also put peripheral. Well, they're not really peripheral, but they're kind of dispersed. Let's put that. But small temporary vacuoles, if any. Forget about this. You can just swap these things. You can just swap those things. These, forget about. This, know these. And you guys already know them because you were the ones that told me that it has glycogen granules versus starch granules. So this is just the same thing. This is just saying that this has starch granules. And this is just saying that this has glycogen granules. And then this last thing, why does the plant cell have a fixed shape?
Because of its cell wall.
Cell wall, perfect. All right, so let's deal with the types of solutions now. So as before, every time I'm gonna start dealing with any compound word, I'm gonna try to break it down. So you have 3 different types of solutions. What are they, guys?
Hypertonic.
Hypertonic, isotonic, and hypertonic.
What's the hypertonic solution?
Concentrated with solids.
What's an isotonic solution?
Equal amount of solids and solvent.
What's the hypotonic solution?
It's concentrated with solids.
All right, so we're talking about types of solutions. We're comparing one type of solution to another. So when we say all right, so I have the meanings of the words on the side, right? Tonic just means concentration, of course, but you guys know that, right? So hypertonic, what we're trying to say is that the solution that we're dealing with is more concentrated than another solution. Isotonic, the solution that we're dealing with is the same concentration as another solution. Hypotonic, the solution that we're dealing with is less concentrated than another solution. Hyper just means more than, iso just means the same as, and hypo just means less than. So again, the words just tell you what the things mean, pretty much. So when we place, let's try the question on that. I guess I didn't. All right, I guess I didn't. So that's fine. What happens when we place an animal cell in distilled water?
Swell and burst.
Swell and burst. Anybody know the fancy word for that?
Okay, so
lysis. It's called lysis. Lysis just means to split. Anybody knows what happens when you put a cell in a hypertonic solution?
It shrivels.
Becomes flaccid.
Yeah.
Anybody knows the fancy name for that?
Plasmolyzed.
No.
Recessive?
Oh, plasmolysis. Sure, in plant cells. We call it plasmolysis. We call it in animal cells.
Flaccid.
We call it crenation. So I'm not saying that you guys need to know that, but it's cool to know. So osmosis. Someone read the definition of osmosis, please? Or give me a definition of osmosis.
Osmosis. Water moves from an area of high water potential to low water potential across a membrane until equilibrium is established.
All right, what does that mean?
Sorry,
What does that mean?
There water moves where there is
So it means that it is moving from an area where water is highly concentrated, means that there is more water in that area, to an area where there is less water.
What's water potential? Anybody know what water potential is?
Not sure.
It's like it's the same thing as saying water concentration, but don't say water concentration. Water can't be concentrated. So we say water potential instead, because everything just has to be complicated for some reason. But it's almost as if you were to say water concentration, right? So if you want to think about it that way, you can think about it that way. What's the important thing here is that it's across a membrane, of course, until equilibrium is established. What's equilibrium?
Equal amount.
Equal amount of water on both sides.
Yeah, equal concentration on both sides of
the cell
of a membrane. What kind of membrane? What do you typically call it? You typically call it like a partially permeable membrane, right?
Or
so
said that we should have
selectively permeable, but we typically say selectively permeable when we're talking about like solutes and stuff like that. But yes, selectively permeable, partially permeable, anything, it's fine. Can somebody please describe to me what's happening in both the animal and the plant cell in the different solutions and why? I'm calling somebody. If nobody's gonna answer, could somebody answer, please? So I don't have to call anybody.
What's the question again, sir?
Explain what's happening to the plant and the animal cell in the different solutions and why it's happening.
Due to the strain of plants or due to the absence of a cell wall.
So tell me what you're seeing.
The plant cell is
has taken a lot of water and so it swell and bursts.
The plant cell?
Animal cell.
Animal cell.
The animal cell is taking a lot of water. Because describe the concentration outside. The concentration outside of the cell. Is it more or less concentrated?
More, less, sorry.
Less concentrated.
Less concentrated. Describe the concentration inside the cell. I'm gonna start picking on people.
Hypertonic.
Describe to me what's happening for the isotonic solution. Could let's see, could Chantel, could you describe that for me, please?
Sorry, repeat, please.
Could you describe to me what's happening for the isotonic solution for the plant, for the animal cell side?
Sorry, no, I cannot.
All right, so when we look at the diagram, what we're seeing is that nothing is really happening, right? So isotonic solution, the concentrations inside and outside are the same, right? So if the concentrations inside and outside are the same, we're in equilibrium. And osmosis only occurs when there is no equilibrium. So if we're at equilibrium, there will be no net result. You're not going to see any observable change in the cell. Cool? That's how I would describe it. Let's deal with a hypertonic. Could Joel, could you describe that for me?
Could you repeat your hypotonic?
The hypertonic. What's going on in the hypertonic
that's,
animal?
I think that's when the concentration of the cell is higher than water. Wait, let me think. Higher than the outside solution.
The concentration of the cell?
The outside solution is higher than that of the cell. So there'll be a net movement of water outside to the out or outward to achieve equilibrium.
I'm very happy that you said the word net movement, by the way. That's good. That's very good. Can you describe to me what happens for the plant cell with a hypotonic solution?
So for the hypo,
yeah.
Or hypo, there's the solution. The solution is less concentrated than what the cell is. So this outside solution has more water molecules. The water molecules will move into the cell, and the vacuole of the cell, the part which contains water, will expand, and that will create kind of a turgid pressure in making the cell more turgid, as it won't burst because it have a cell wall.
So yeah, perfect. If you want to make it, you can say it increases turgor pressure. Turgor pressure. So instead of saying it more turgid, so turgor pressure is increased because it's a pressure, right? Can Jabari, can you tell me what's happening for the isotonic solution for the plant?
It's equal pressure on both sides, so there's no thing happens.
Yeah, there's no gross change, right? Perfect. Could Brianna describe to me what's going on for the hypertonic solution with the plant cell?
So on the outside of the cell, the solution is hypertonic, and inside it's hypotonic. So because it wants the overall solution to become isotonic one, it releases water, and so that's why it becomes shriveled.
So, all right. So why is it shriveled?
Because it's releasing water.
And what do we notice about the vacuole?
It becomes smaller because that's where the water is coming from.
What do we notice about the cell membrane?
It shrinks as well.
What do we call that?
Plasmolyzed.
Plasmolysis. All right, perfect. Actually, yeah, perfect. What do we notice about the cell wall?
Stays the same.
It
stays the same.
Maintains its shape. Perfect. All right, guys. So you guys have it, honestly. I don't think there's anything more for me to add. Can someone just briefly go over everything like really quickly, like a minute and a half? Anyone? Brianna?
Repeat what you said?
Can you just go over everything just quickly?
So the first animal cell, it's swollen because it's taken in water, because the solution outside is hypotonic. The second one,
it's swollen. What do we notice about
no, it becomes rupture.
Okay.
For the isotonic solution, there is movement from going inside and outside of the cell, because water is always moving. For the hypertonic one, no.
Tell me more about the isotonic. What do we notice about it? Is there any major change that we can see?
No.
Okay, good.
Hypertonic, the cell becomes shriveled as it shrinks because it's releasing water. For the hypertonic solution.
I'm not gonna force you to do the whole thing. I wouldn't. I'm not so wicked. Let me look who I'm gonna be wicked to. Ashley, could you tell me about the plant cell, please? Honestly, I don't know why I'm calling people out. I would hate this. But can I say? Yeah, Ashley, can you tell me about the plant cell, please? All right, Joel, I think I called on you before. Joel, can you tell me about the plant cells?
Plant what?
The plant cells. Can you describe what's happening with the plant cells?
I think because water is going out, the cytoplasm is pulling away from the cell wall.
So start with the hypotonic. Start with a hypotonic solution.
Oh, hypo. Oh, sorry, did answer this a while?
Yeah, just answer again. It's cool.
All right. For the hypo, water is moving into the cell. Because water is moving into the cell, the vacuole, which is the organelle which hold water in the cell, increases. This increases causes it to exert turgid pressure and causes the cell to become rigid or turgid. And because it have a cell wall, it's not going to burst because of this pressure.
Perfect. That's the word, turgid pressure. Next, isotonic.
Isotonic, because both the concentration are similar, there's no net movement of, no net exchange of water molecules.
I like that. Hypertonic.
Because the outside solution is more concentrated than the content of the cell, water molecules move outward and hence causes the cytoplasm to pull away from the cell wall.
Cytoplasm?
Yes, sir. I said the cytoplasm that like a pull away from the cell wall. No? Oh, Jaja.
Try again. All right, let me tell you why they call it plasmolysis. So they call it plasmolysis because we call it cell membrane right now, but we can also call it the plasma membrane. So what's shrinking is the cell membrane or the plasma membrane. So it's a cell membrane that's actually pulling away from the cell wall. You get it?
Oh, yes, sir. Yes, sir.
Yeah. So the plasma doesn't just appear out of nowhere. It's just, yeah. You guys, I wouldn't expect it to have just known that out of nowhere. But yeah, so continue.
Yeah, because of this loss of water, the cell in essence become flaccid. You know, it's feel, you know, flaccid.
On to the vacuole.
The vacuole becomes smaller too.
Yeah, perfect. Describe the turgor pressure. Is it high or low?
I would assume it's low because you lose water.
Yeah, perfect. Importance of osmosis in living organisms. I saw a question like this. So just kind of pay attention. Ensures that our cells maintain turgor pressure, turgidity. In animals, helps to absorb water from the intestine to the blood, from the intestinal cells anyway. Maintenance of the cell shape, it does do that. Plants, prevents wilting, and aids in nutrient transport. I should have put water instead of nutrient per se, because it can't really be nutrient. Well, I mean, when water is being transported, you have nutrient transport, but put water. Aids in water transport. Cool? Everybody good so far? These are just things to know. It's not something I can explain really. It's just stuff to know.
Okay, sir.
All right, cool. Here's an experiment. So if I was to say design an experiment using potato cubes to show how osmosis works, use that stuff and just tell me what you would do. Yeah, hello?
Oh, sir, could you go back to the previous slide? I wasn't finished, right?
You can take a screenshot here. All right, you good?
Good now.
All right, great. So osmosis experiment. So I just want someone to kind of like give me an experiment that you could use to kind of like examine osmosis with the stuff that you have here. Just like tell me give me like kind of like some idea of what you would do.
Sir, put up.
Any volunteers? All right, you guys are
in that one. In distilled water and the other in the sugar. Is it sugar solution?
Yeah, that's one mole. You guys know about moles, right? You study the chemistry. So mole just means concentrated sugar solution. So it's concentrated. So that's how you get the solution concentrated. So yeah, continue.
So sir, put 3 of the potato cubes in the distilled water and then the other 3 in the concentrated sugar solution. And then I just know the change.
You have 3 potato cubes testing for one thing and 3 testing for another thing. So what's the purpose of the measuring cylinder?
Sir, maybe for the liquids.
All right, so if I was to plan an experiment like this, how I would think about it is you have your 2 extremes. You have your distilled water, which is your most hypotonic solution. You have your 1 mole sugar solution, which is your most hypertonic solution. But like you guys know, when you have your juice and the juice is too sweet, if you just put a little water on it, then you can stretch it. It can become a bit more fresh, right? So if I have my 1 mole sugar solution and I add a certain amount of water to it, it's gonna dilute it by a little bit, dilute it by a little bit, dilute it by a little bit. So, or if I add less water, it's gonna concentrate it. So I want different concentrations of solution. So what I'm gonna do is I'm gonna pour a certain amount of distilled water and a certain amount of sugar. So all I'm doing is just adding water to sugar and then dropping a different potato cube in there. So I'm gonna make different concentrations of solutions because I want one that's isotonic, one that's kind of as close as possible to the actual concentration that would be in the potato cube, right? Because if I want to test all three, if I just use distilled water and 1 mole sugar solution, where am I going to test for the isotonic, right? You guys following so far?
Yes, sir.
Yeah, so like, yeah, if I have a big jug of sugar water, I'm going to want to dilute that over little by little, add more water, so I can test for more stuff. So in the first test tube, all right, let me just try. In the first test tube, I'd probably put distilled water. And in the last one, I'd probably put my 1 mole sugar solution. And then going through, I'd add maybe this amount of distilled water, and then with like this amount of sugar solution, and then I'd add more sugar going along. All of them would of course have the same height, but you understand what I'm saying? I'll add considerably less and less water. So as we go along, I'm getting more concentrated. So say this is 1 mole, I dilute. So we can hypothetically call this 0 moles of sugar. Maybe call this like how many are there? 1, 2, 3, 4, 5, 6. So, and this is 1, so 0.2, and then make this like 0.4, then make this like 0.6, then make this like 0.8, and keep concentrating it all the way up to 1. And then even somewhere along here, say call it here, this is probably the isotonic solution. Of course, they're all the equal weight. I have to measure them all beforehand, right? Measure them all beforehand. I drop them out, I take them out, and I weigh them, right? And what would I expect for the potato cube that's in the distilled water? Is it gonna weigh more or less?
More.
Or
What would I expect for the one that's in about the 0.4 to 0.6? Is it gonna weigh more or less?
probably the same.
About the same, yeah.
About the
What would I
same.
expect for the one that's in the 1 mole sugar solution? More or less?
Less.
Yeah, you guys got it. See, you guys have it, man. So we can move on from there. We're actually going pretty well, so we can deal with the next topic. Effects of climate change. So can someone read, please? Abigail, can you read, please?
Okay. Effects of climate change. 1. Excessive amounts of gases such as carbon dioxide in the atmosphere have resulted in increased trapping of heat energy around the Earth, causing higher global temperatures, in brackets global warming, similar to that which occurs under greenhouse conditions. What are the benefits of growing plants under greenhouse conditions?
All right, can somebody tell me that? Anybody does the agriculture? Or you don't have to do
Sir, you can increase the levels of carbon dioxide, which increases the rate at which the plant grows.
Give me something else.
You get to control the limiting factors of the plant.
Which are?
Carbon dioxide concentration, water concentration, humidity, wind.
You're missing a major one.
Temperature.
So grow plants in greenhouses because we can control the temperature. Oh no, it's cool, man. It's cool.
Wait, so what was that?
Oh, no, I'm about to write it. So, what are the benefits of growing plants under greenhouse conditions? So, I don't know. I don't know if we use greenhouses in Jamaica. I'm not a big horticulturalist, so I don't know. But I know that they use them in like Europe and those places during the winter. Why? So, the snow is falling outside, but your plants are inside growing just fine. Why? You get to control the internal temperature, which is essential for plant growth. Yeah, so plants aren't going to grow in the snow, but with the greenhouse, you get to control plant temperature. For the next one, can somebody suggest 2 reasons?
Control the concentration of carbon dioxide.
Is not generally desirable. Suggest 2 reasons.
Oh no, I thought you had to give like more than one benefit for like growing plants.
Honestly, I don't know any more benefits for greenhouses. I don't know any more benefits for greenhouses. The most thing I know is that they grow them in a greenhouse because you get the temperature control. But as I say, I'm not a big plant guy. I don't do plants, I do people. So yeah. Well, as far as I know, it's mostly about temperature and maybe like adequate growing conditions. Because if you just left the plant outside, it's not like carbon dioxide is really a very big limiting factor, you know? There's air all around. Yeah. So just 2 reasons why global warming is not generated. Oh, I saw places getting hotter. Increased temperature. What does the increased temperature cause?
Melting of ice caps.
Yeah, melting of the polar ice caps.
Rising sea level, flooding.
Rising sea level. Perfect. If the sea level rises, what happens?
Flooding. Lowland areas.
Yeah, perfect, guys. Perfect. Give me another reason, please.
Sir, you increase the acidity of the ocean.
Ocean acidification. Perfect. For people that don't understand why that happens, the hotter it gets, tsunamis are maybe hurricanes. I don't know how tsunamis cause. Maybe
because
hurricanes.
severe hurricanes also.
Yeah, hurricanes worse. But yeah, ocean acidification. The hotter something gets, the more stuff can get dissolved in there. So, all right, I don't want to get too kind of like, you guys, if you don't understand, that's fine. But the hotter something gets, the hotter a solution gets, the more stuff can get dissolved in there. So if you have a regular room temperature and you try to dissolve some sugar in there, and you have some sugar that's not fully dissolving, if you heat up that pot, the rest of the sugar should dissolve. Everybody agree?
Yes,
Everybody agree? Like, have you ever done that before? I'm talking about just dissolving sugar.
Yes.
Or if you're mixing drinks and it's not all dissolving in, you just add hot water to it. All right. But in general, so yeah, that's a thing, right? So next time you're making juice, yeah, that's a thing. So with respect to the ocean now, so that body of water, again, it's the exact same thing. In that body of water, you have gases like carbon dioxide being dissolved in there. So when carbon dioxide is dissolved in water, it forms a weak acid. That's just, all right, it forms a weak acid, right? So the more carbon dioxide, if it's hotter, and it's the same thing with the sugar, you can dissolve more sugar, you can dissolve more carbon dioxide. So if you're dissolving more carbon dioxide, you have more weak acid. I think it's carbonic acid. You have more weak acid being dissolved within the ocean. So you're just increasing the acidification. That's not very important to know, but I mean, if you want to know, that's all I'm telling you. Just if you want to know, it's not a thing you need to know, right? Increase in temperatures. All right, so just one way in which global warming may be advantageous. You guys have to think about this because nobody ever asked that.
Does it cause more rainfall?
No.
Sir, is it because that cooler countries can become more warmer or warmer?
Yeah, that's what I would have said. Perfect. Increases habitable areas. For humans anyway. And other animals.
Sir, and plants can grow during the winter.
Yeah, sure. But not it changes the climate per se. It changes the climate. So
Like
it
rainfall.
just makes the area less hostile. Rainfall, yeah, it probably would increase the rainfall. Let me just clarify this about the acid. Now, it doesn't later become a strong acid, but if you have a bag of weak acid, then you just have more acid. So it's just more acidic. It's not that it's suddenly strong or suddenly weak. If it's weak, it can become strong, and if it's strong, it can become weak. You just have more of the weak thing, or you just have more of the strong thing. Cool? Cool. All right, next thing. So this is what we're supposed to cover for climate change. So climate change, can somebody read this, please? Joel, can you read this, please?
Refer to the average increase in the global temperature due to the increase in the greenhouse gases, causing a rise in sea levels and ocean acidification.
Can you give me an example of a greenhouse gas?
Methane.
Methane.
Can anyone give me more examples?
Chlorofluorocarbons.
Chlorofluorocarbons. What else?
Water vapor.
Nitrogen
Water
oxide.
vapor. Nitrogen oxide. Probably. Yeah, probably. All right, greenhouse effect.
But please, not the previous slide. Wait, not this. Yeah, this one. Okay, you can continue.
But yeah, water vapor. I'm glad you guys got water vapor. So as again, gases such as methane, carbon dioxide, and water vapor trap the sun's heat on the earth, increasing the earth's temperature. So this is how the greenhouse effect works. Sun is giving its energy, it's coming down like this. A lot of it is supposed to bounce off the atmosphere, but because we have a lot of greenhouse gases in the atmosphere, it just ends up being trapped, and yeah, and it goes up, down, up, down, up, down, and it's just radiating right back to the earth. So it's just not escaping. So we just get hotter as a result, right? Just like in a greenhouse. So we're moving on. We're actually moving so quickly, guys. You're doing very well. You guys want like a 2-minute break?
Yes, sir.
No.
All right, guys, make up your minds. So
Yes, sir.
type
Yes, sir.
Type it in the chat if you want it. All right, cool. I'll give you guys. All right, let's split the difference. You guys get a minute-long break. So enjoy. Yeah, what's your question? I could. I really like chemistry. I really enjoy it. Is it CSEC chemistry? I say I don't really enjoy chemistry. It's not very fun or very interesting. I could though, but there's a chemistry class, so that should be coming up. That should be good. But yeah, I don't like chemistry. I don't like. Well, I mean, I said I don't like it. It's stuff I have to use in like everyday life, but I still don't like it. I prefer not to do it. But yeah, it's fine.
Okay, sir.
Yeah. But if you need help and I can help you with it, I'll help you if you just message me. All right. So you guys done with a minute, which I actually gave you like 2. You guys good? If you're ready, sir, ready. Nobody's ready. All right, I'm ready. Ready. So let's continue. All right, cool. Christian. Only person that's on my side. All right. So all right. Let's do blood component, sickle cell anemia, and genetic diagrams now. So scientists establish relations. All right. Can someone read, please?
Scientists establish relationships among individuals using their knowledge of genetics. Construct a sequence of genetic diagrams to illustrate how scientists can use knowledge of genetics to show how a couple with blood types A and B, respectively, can produce a baby with blood type O.
Okay, guys, let's do this. I'm gonna just say I have no idea how to do this, so you guys have to walk me through it, right? So how do we start making a genetic diagram?
So you put the parents' genotype at the top. So.
What genotype are we crossing?
So what did they say? Oh, AO.
What?
AO and the BO.
And this is how I want you guys to write your genotypes. Okay. This is how I want you guys to kind of like do it the way I do it, because I kind of do it. I think I do it kind of methodically. So it's not like you're gonna miss out on any steps or anything like that. So if you're not sure how to do this, just kind of like pay attention. So what's the next thing I'm gonna do?
Then you're going to cross. So the A will go to the B.
How do I draw my square? What?
You can also use Punnett square for the right.
I have to draw my square.
See, sir, I thought you were just crossing, not Punnett. Okay.
Nah, I always have to draw my square.
Okay, sir. So
I can't do it out of my head. Can't do it out of my head. I have to draw my square. So how are we gonna draw a square? All right, let's draw the square.
And then put the 2 align in the middle.
I think it's 9 boxes. Or it's not.
No, just you guys doing. I didn't know if you guys did crosses with 9 boxes. Do you?
No.
No.
Sir, repeat
you guys don't do crosses that result in like 9 results, do you? Just the 4.
No, sir, only 4.
Okay.
I put the AO on the parents' actual thing in the box.
All right, guys, which one do we think is the mommy and which one do we think is the daddy? AO or BO?
Read, sir.
AO for mommy.
Which one is the mommy and which one is the daddy?
AO for mommy.
AO for mommy, BO
Yeah.
for daddy.
Yeah.
Typically, I go male then female, but I write when I write to my parents. All right, male. All right, male then female. But it can literally be anything. If they don't tell you, it can be anything. But just do male then female. So what's the symbol for male? All right, I'm gonna draw two symbols. Which one is male and which one is female?
Second one is
The
male.
arrow is male and the cross like
The first one is female. The
Okay,
second
cool.
one is male.
Good. So if this is male, which one goes on the top, male or female? Which one goes up here?
The male, since the male is AO.
So, all right, so I put, what do I put here? I put A here and then O here, is that correct?
Yes, sir.
What do I put after putting my after putting my symbol, what do I put here? I put O here?
BO.
Okay, BO. Right. All right, so when I'm crossing them, what do I get in this slot?
You get AB.
AB.
So when you're writing, you start up here and then you go down here. So yeah, AB. So what do I get here? Actually,
AO.
no. Here? Oh, AO. Yeah, AO. When I'm doing it here, what do I get?
BO.
BO. What do I get here?
Then you have O there. Or, yeah.
So now we're gonna write our results. Results. When we're writing our genotype, what's our resultant genotype?
AB.
How many ABs?
1 AB.
1 AB.
And
colon.
one AO.
1 AO colon.
1 BO.
1 BO colon.
And 1 OO.
1 OO. And what are our phenotypes?
Blood group
Group
A.
AB. What's our next phenotype?
A.
1 group A.
Group A. What's our next
And
phenotype?
1
1 BO. What's our next phenotype?
1 group O.
1 group O.
So you can
Perfect.
have B.
Honestly, it's not BO, it's just B.
Okay.
BO and B are the same thing, by the way. BO, BB, same thing. It's blood group B. BO is the genotype. B is the phenotype. So yeah, so how did the two parents produce a child with blood type O? They each had to they had to be AO and BO, right? Everybody good?
Yes.
So you normally like 25% chance.
Pardon me?
Like you had like 25% chance that the baby would be AO.
Yeah, 1 out of 4. Yeah, perfect. All right, so we finished that question. So let's do this one now. Can someone read, please?
A tropical plant species produces flowers that are either purple or white. The purple color is found to be dominant to the white. Using P as a symbol, state the possible genotypes of the purple flowers.
All right, so what are we thinking?
Capital P for dominant, purple flowers.
And common P for white.
All right, so what's the genotype of the purple?
PP.
Capital or common?
Sir, could I have both capital?
Both capital. What else?
And a capital and a common.
Perfect. Capital and common. I'm gonna get into that after this. So of course the explanation is coming, but yeah. Plant breeder. All right. Plant breeder obtain flowers purple successive crops. However, one crop obtained a few white flowers. Using a genetic diagram, illustrate how we got white flowers in a crop of the same plant species. So how are we gonna do that?
Sir, probably.
What do we start with?
The genotype.
genotype. So what are we gonna cross?
So you could cross that two heterozygotes, heterozygotes, and
What's the mother?
so it could be heterozygotes too.
Two heterozygotes. Okay. All right. So what next? So let's draw our square. What goes on top?
Male.
What goes here?
Female.
Female. All right. So if we're crossing big P little p versus big P little p, then what are we gonna get here?
Sir, two capital P's.
Two capital P's.
At the bottom. Oh, yes, sir. Over there, have a capital and a common.
Here.
A capital and a common also.
And what do we get here? The last one.
Two common P.
Two common P's. So now we're writing out our results. Now we're writing our results, right? So what are our results? So our genotype of our results. What's the genotype of our results?
One, two, the two capital
P, capital
and then two big P, common P.
Two big P,
capital P,
and
and
one.
then
one common.
Recessive.
Yeah, perfect. What's our phenotype?
One purple
flower.
One purple flower.
No, it's three purple flower.
Three purple flowers.
And one white flower.
and one white flower.
One white. So here's our white. Here's our white. Here's our white. Perfect. So next, genetically identical offspring sometimes show difference in phenotype. Suggest why such differences may occur. That's our question, by the way. Genetically identical. Note that.
Sir, because of variation.
Genetically identical. Do you mean crossing over in meiosis? Is that what you mean when you say variation?
No, as in like the environment that they're in.
Environment.
Plus their phenotype.
So
Environmental factors play a role in phenotype. So they might have just been exposed to different environments. All right. Next.
Can you go
allele
back
versus
to
gene. What's that?
Okay, thank you.
All right, definitely. So what's an allele versus a gene? Define gene first.
A gene is the basic unit of heredity that codes for a specific characteristic.
Yes, but it's also just a segment of DNA that codes for a specific characteristic. But yes, what you said is perfect. Right, what you said, but for everybody else, it's just a segment of DNA that codes for a specific characteristic or a specific protein or whatever. But it's just a segment of DNA. If you put a segment of DNA, I don't think they can give you it wrong. But for gene, who said that, by the way?
Me, sir. Joel.
Yeah, great answer. Of DNA. Repeat the answer.
I said the basic unit of hereditary that codes for a specific character.
The basic hereditary unit. So you're just saying DNA, or you're just saying a segment of DNA. That's all that's saying. that codes for a specific characteristic. Yeah, perfect. You're just saying the same thing I'm saying. But yeah, that's a great way to put it. I really like that. Just that codes for a specific characteristic. Or it's hereditary or something like that. Hereditary, right? I think that's the adjective. So what's an allele?
A trait.
A what? I couldn't hear that.
So I was saying a specific trait or single trait.
Yeah, but give me more than that.
Form of a gene.
Yeah, alternative form of a gene. Perfect. Yeah, alternative form of a gene. So
Sure,
just a different form of a gene.
So that's it. That's just it. That's it. That's just it.
Yeah, that's just it. That's just what it is. That's literally just what an allele is. It's just a gene. It's just a different form of a gene. Yeah, there's not much to these stuff. Yeah. So what's phenotype and genotype? Define genotype first. You see, it's funny. It's funny. They gave you the one. You shouldn't define phenotype first. Define genotype first.
The genetic makeup of a.
Perfect. Define phenotype.
The physical aspects due to
Perfect.
the genotype.
Literally perfect. You don't need much. Link to what? Link to?
The genotype.
The genotype. See, perfect. That's literally a perfect answer. That's literally a perfect answer. If you want, you could put coded by proteins. But that's like a higher level answer. So I'm putting it in brackets. Dominant and recessive. Define dominant first.
An allele that determines the phenotype of the organism.
Give me that in a different way.
It overcomes other.
Who's saying that? I think Maxwell, I think you were saying that. Yeah, just give me that, what you were just saying.
So I was saying that dominant allele overcomes the other traits, so it's the one that is seen.
Yeah, perfect. So that's what I would put anyway. I'd put the dominant allele is the allele expressed always. It's always expressed, but I just can't bother with English sometimes. Always expressed when present. Always expressed when present. So what does that mean? You have two big P's, it's always expressed. You have a big P, a little p, it's always expressed. You have no P's, can't be expressed, it's not there. So all I say is that it's expressed when present. So I don't know how else per se to define it. What would you define a recessive idea as?
An allele that is only expressed when the dominant is not present.
That's exactly how I would define it as well. Expressed in the absence of a dominant allele. I don't know why I spelled it that, but yeah, that's exactly how I would define all of these stuff. I'm not gonna really overthink it. These are probably not textbook definitions, or maybe they are. I don't know. But it's just things that make sense. So when you're in an exam and you think, what is this? Yeah, this is what should come to you, right? So I want you guys to be able to think like that, of course. So let's continue. So can someone read, please?
Black fur color is controlled by a dominant allele. Brown fur color by its recessive allele. E. This characteristic is not sex-linked. Give the genotypes of the parents and offsprings of a cross between a black male and a brown female that produces half black offsprings and half brown offspring. Show all the steps in the working of this problem.
You see that, show all the steps, but we know how to show all the steps. So how do we start?
Genotype.
Parent genotype.
So what's the genotype of our parents?
Sir, heterozygote.
So give me the heterozygote combination.
Have
B
and B, capital B, small e, and then
small. It's not small e. The recessive allele is a capital E. So you have to pay attention. It's not just a general thing where you just put a common letter. What they said that the recessive allele is a capital E. So if you put a small e, you get it wrong.
Oh, okay, sorry.
Yeah. So what's the next thing? What's the
Same. BE.
And our female is a BE as well. So, all right. So let's draw a box, right? So right here we have BE. Right here we have BE. Of course, making things neater. Doing the examination. For or what do we have at the top? Male. What's the symbol? This. What's the symbol for female? This. So what do we get here?
BB.
Next box.
BE.
BE.
Next box.
BE.
Next box.
EE.
All right, guys. So what do we have? BB, BE, BE, BE. So what do we do next?
Oh, sir, EE.
How you have BE for the last one?
Yeah, EE. That's my bad. I'm sorry. EE. So what do we do next?
Show the result.
Results. What's the first thing we put?
BB.
What was the first thing that we put? What's our next heading?
Genotype.
Genotype. What are our genotypes?
one BB.
1 BB.
2 BE.
2 BE.
And 1 EE or the 1
1 EE. What are phenotypes?
B, B,
Black
E,
fur,
E,
one. Black fur.
B, and B, E.
3 black fur, sir. Or 1.
1. Pardon me, 3 black. I heard somebody say 3 black.
And 1 brown.
And 1 brown. Is this what the question asked?
No, sir.
You know this, I'll let you guys do it. Yeah. So this is the part that you guys were ignoring. So it's a black male and a brown female.
You know, I was
This is
wondering
female, brown.
you're the one teaching, so
Yeah, I'm not just gonna randomly correct you guys when you're doing it. I'm gonna follow you guys through. You guys give me an answer, I'm gonna follow it through. But yeah, so what would you need for a brown female?
EE, capital EE.
So what it should be, the cross should be EE and what kind of male?
B.
If we use BB, all of the answers, all of the results are going to be BE.
I can use the heterozygote, the BE.
We'd have to use a heterozygote. So I'm not going to do the whole process again, but you guys should do the whole process again. I'm just going to do this quickly. BE, EE, just to show you. BE, EE, BE, EE. BE is what? Black. EE is what? Brown. So we get our half. But yeah, notice that I did not correct you guys. Why? Because you guys need to be sharper than that. Yeah? You guys need to be the ones correcting me. But yeah. Anything else? But I mean, the working is good. And you guys would have gotten to the end. You notice you guys would have gotten to the end, gone back over the question, realized, oh, that's not what they asked for. So something, I have to change something. So you'd have ended up changing your genotypes anyway. So you would have eventually gotten the right answer. So you notice when you follow it through to the end anyway, you end up in it, because some people just put BE times BE and it says, oh, what am I supposed to get? A half black, half brown. So I'm just going to put something that would give me a half black, half brown. And then they would end up getting it wrong because I didn't work it through. But because you worked it through and you worked it through to the end and you realize, oh no, I got the wrong answer. So you know that you have to go from the top and work it back through. So that's why you always have to work it through methodically like this, right guys? So everybody good?
Yes, sir.
All right, great. So next. So hemophilia, you guys, the X-linked conditions, right?
Yeah.
Yes, sir.
All right, great. So hemophilia is caused by an X-linked recessive gene. Two parents with a normal phenotype have a hemophiliac child. Use a genetic diagram to show how this is possible. So how do I start? I always start the same way, guys.
So you write the parents genotype.
Yeah, I always start the same way. What would be my genotypes?
XH and XH.
What are we doing first? Male. We're doing male first, right?
Yes, sir.
So what's the sex of a male? What's the
XY.
XY? So it's X-linked recessive. So to have a normal male, what's his genotype?
Oh, sir, that would XY would have been his phenotype.
XY is his genotype. His phenotype is male. XY is his genotype. So tell me, to have a normal male, a non-hemophilia male, what does his genotype have to be? It literally has to be. What does it have to be?
X, a capital H and Y.
X, capital H. What do we put on the Y?
Nothing.
Nothing. What? Why?
Because doesn't carry the X, the Y axis cannot, the Y axis. No, the Y gene cannot carry the trait. It has to be an X.
It's too short, basically.
Yeah, it's too small. You guys know how big a Y chromosome is? It's very small. Very, very small. So what are we doing for a female? What's the genotype of a female? XX. So this lady is normal, right? She's phenotypically normal. what does her genotype have to be for her to have a hemophiliac child?
H.
One person, please. I can't hear.
Capital H and a small a.
Why?
So,
So how is she
because
normal? How is she normal then?
because it's not really
Is she a carrier?
she's a carrier, sir.
Yeah, but remember what we said? Once there's a dominant there's a dominant there, it's gonna be expressed. So yeah.
Yes, sir.
So this is our test cross, right? We're just doing male and then female because why not? I don't think it actually matters. So we're doing male, so we're doing our males first. So when we look at our X and our Y, we put our X and our Y here. We're doing our female here. We're doing X and X. So what's our genotype on the X for the male? What allele is on the X for the male?
The capital H.
Capital H. What allele is on the Y?
Nothing. No. Oh.
Nothing. So I just put a 0. It's a 0, not a O. It's a 0.
Okay, sir.
So what is on the X for the first X for the female?
The H, capital H.
What's on the second X?
Another H.
Common H. So when we do our cross, what's going in the first box?
X, H, capital H, capital H.
Oh, you can just fill this out from right now, by the way. XX, what's the next thing? What's here?
XY.
XY, what's going here?
XX.
XX, what's going here?
XY.
XY. So let's do our genotypes now. So for the first X, X big H and X.
H, capital H again.
Capital H.
Right. So for this male, what's his age?
Capital
H with a
For this female, what's her first age?
capital
H, then a common H.
Common age. What's this male's age?
common
Common H.
All right. So what's next?
results
Results. What do we write next?
Genotype.
Genotype. What's our genotypes?
One.
X,
XX with the capital H.
What else?
1 XX capital H common H.
What's next?
And then we have
One
capital H. And then
Yeah.
1 XY again common H.
All right. So what next?
Phenotype.
So what's our first phenotype?
Normal female.
Normal female.
And
One
then
normal female. Next.
carrier female.
One carrier female. Next.
One normal male.
One
Normal male.
male. Yeah.
And
One
hemophilic.
hemophilic male.
One. You sure he's not normal?
Sir, it's
Am I
hemophilic.
lying?
This is
Nobody
the
got
common H, the carrier thing.
One hemophiliac male.
No one is hemophilic.
Or we can just say affected. One affected male. So how can two phenotypically normal parents have a hemophiliac child? Mother has to be a carrier. Father has to be normal. Can you have a carrier male?
No, sir,
Yes, sir.
No,
Why?
because the Y,
because only
I
the
feel
They
like
will
you guys
have
got
one
it.
egg.
can carry the common seed.
the
Yeah,
Y
you guys
cervix.
got it. I don't even know what you guys are saying, but you guys got it. From you mentioned the Y chromosome, you guys are good. Because there's no other X to mask the hemophiliac gene, right? So like in a female, you can have one dominant X and one recessive. You can't have that in a male, right? So yeah, so that's why people do like genetic testing and stuff like that before they go and find a partner. Because if you go and you have a child with someone with a completely normal male, you could end up having a hemophiliac child. Anybody knows what hemophilia is?
One with the blood, clot something.
The blood is unable to clot.
Anybody knows what philia means?
Repeat.
Anybody knows what philia means? What does the suffix philia mean? Philia just means you like something. Philia just means you like something. What does hemo mean?
Is it blood?
Blood. You like to bleed. You like to bleed. That's how I think about it. You just like to bleed. So it's a problem with your clotting. So your blood can't clot. So you just bleed. You get a cut, you bleed out, you die. Yeah, that's how that works. And that's why you should not marry your cousin, because stuff like that goes down the line if you end up doing that. So when you look at like the royal family and stuff like that, that's the kind of examples that they give for like hemophiliacs and stuff like that. Yeah. So let's continue. So we're just going into the part where we actually talk about it. So you have different types of the different blood types. You have A, B, and O. A and B are codominant. So that means you can have the expression of A and B, or you can have A by itself and B. So they're both dominant. That's all it means. So when they're both there, they will both be expressed. That's all codominant means. O is the recessive one, right? So you have to have two O's for O to be able to show up, right? So I don't know if you guys have to know about antibodies and antigens.
I don't think so.
Sure, sir.
All right, cool. Let's just skip that then. Or do you guys want to know about it? Let me know.
No, move on, sir.
Oh, okay. All right. Let's just touch on it really quickly. If you're blood group A, you have an A antigen. If you're a blood group B, you have a B antigen. If you're blood group A, B, you have an A and a B antigen in your blood. If you're blood group O, you have no antigens in your blood. So antigens are just what you produce. So my blood in my body has an A antigen. That means my blood is saying, hey, I'm an A. So in my body, I have anti-B. Antibodies have anti-B because I have A. So I need to be on the lookout for B, because if I have A, I wouldn't have anti-A. I wouldn't have anti-A. I'd be destroying my own stuff. So I need to have anti-B. If I have B, I need to have anti-A. If I have A and B, I can't have any antibodies. Why? Because I'll just be killing my own cells. If I have no antigen, so if I have O, then I need anti-A and anti-B, because if I just suddenly start having A and B antigens in my body, it can't be mine because I'm not producing that. I have none. But that's just a little simple rundown of blood types. So that's how blood types work when you go to the blood bank and you do need blood transfusions. And that's how the blood transfusions work. So the types must match. No. Think about it. If you have A antigen and an A antibody, you just have one bag of Yeah, all that's happening is that your own body is killing your own blood cells. That's all that means. Sickle cell is a whole other disease. Sickle cell is a whole other disease. So blood transfusions, the type must match. That's just what's happening here. I think, do I have anything on sickle cell? I don't know. A and B are, do you guys have to know about sickle cell, by the way?
Yes, sir.
What do you have to know about it?
It says sickle cell anemia on the topic something.
Oh, yeah, all right. So yeah, it's just the stuff I have. All right, good. So A and B are codominant, so they're equally expressed. So that means is that you can have somebody with with A, B. You can also have somebody with just A. You can have someone with just B. And the O is recessive. Of course, we know what that means. So which alleles are dominant? A, B over O. Which alleles are recessive? O. Which alleles are codominant? A and B. Right? So everything just cool with that. So here's our sample Punnett square again. So this is just we already did some Punnett squares for these, right? So should be good. Genetic diagrams. So again, gene, a segment of a strand of DNA which codes for a specific protein. Codes for a specific protein is the same thing as saying codes for a characteristic, because I know that's what you were saying. Codes for a characteristic. Codes for a protein is the same thing. Allele, variation of a gene, we did that. Types of genes, dominant and recessive. Dominant, always inherited and expressed when present. And we typically express that with a capital letter. Recessive, expressed in the absence of a dominant gene. And you guys already know this stuff, right? So this is just a sample cross. What would this person, well, let's just go to the next thing. I think I have it next. All right, so I have males on top, females on the bottom. So if you guys don't remember which one is which, this one, which one is this one? Male or female?
Female.
So yeah, the female one, it kind of looks like a mirror, right? No, it's not typical. It won't be color coded in your exam, but it looks kind of like a mirror, right? You know, like how girls like hold up the mirror and like say, wow, I'm so pretty today. Yeah, so it's like you're looking in a mirror. So it kind of looks like a mirror. So that's how I remember which one is which. So the female one looks like a mirror, and the male one looks like a spear. This is a spear, the arrow part is a spear, and this circle part is a shield. So like a spear with a shield. So if you look, so that's how I remember them. Because I say, when I'm doing my stuff, I need ways to remember the things. I'm not just gonna get up and just remember. Yeah, I'm not just gonna get up and remember like that. So homozygous and heterozygous. So homo, what does homo mean? Same. Hetero means different. Homozygous, you have two types, homozygous dominant and homozygous recessive. So by that same, I mean, so we can see it here, we have dominant RR. Is this homozygous, this RR?
Yes.
Is this RR homozygous?
Yes, sir. Yes, sir.
All right. So this one, the only difference, this one is homozygous dominant, this one is homozygous recessive. Homo, same, they're the same letter. That's all I'm trying to say. They're the same letter. Heterozygous, RR. The dominant feature is expressed. So heterozygous, what does hetero mean?
Different.
Different. So what's different about them?
Common R.
One is dominant and one is recessive.
The capital and the common letter. It's just about the letters, the capital and the common. So yeah, perfect. So let's do this one because I want you guys to make sure that you understand this. So dominant is red wings, recessive is white wings. A red wing female was crossed with a white wing male, and the result was 2 white wings and 2 red wings. What is the genotype of the female? So
RO and RO is that?
Don't we have to start somewhere? So how are we gonna start? How are we gonna start? We have to do our test cross. We have to show our working. So what's the first thing we start with? What do we start with?
Genotype.
Genotype of who?
The male.
The parent.
The parents.
The parent genotype. So what do we want to cross? What are we writing first? What do we have? White wings is recessive. Red wing female, white wing male. So if we're doing male first, what's the genotype of the male? What has to be the genotype of the male? Let's call it P.
R.
Let's call it P. Let's R. Let's use R. Let's use R.
R
So-
for white.
So what has to be the genotype of the male?
2 capital R's.
2 capital R's
Mm.
for the male.
Sir, a capital and a common.
Remember, recessive. White wings is recessive.
RO.
Right wing, white rings is the recessive.
Sir, what is
Oh,
the key?
sir. 2 common R's then.
2 common R's. What was that question?
No, normally there's a key.
Key.
As
Oh,
in like what for represent this and
yeah.
this.
I just forgot to put it in. That's my bad. Sorry. All right. So we're just using R. So 2 R's, 2 common R's. What's the female now? What are we using for the female?
Should use heterozygous, sir?
Heterozygote.
Yes, sir. Heterozygous dominant.
What are the heterozygous? There's no heterozygous dominant. What are the what's the result? And the result was 2 white wings and 2 red wings.
Dominant then, dominant.
Dominant. What kind of dominant?
It's the both of them should be
Can be,
dominant.
heterozygous.
Huh?
They're homozygous.
Homozygous, both of them.
Homozygous,
should be
homozygous or
dominant.
heterozygous? Guys, choose.
Heterozygous.
Heterozygous. Who says heterozygous? Type in the chat.
Sir, I'll say homozygous dominant.
Type in the chat. Type in the chat. Homozygous, hetero. Guys, type in the chat. Don't be afraid. It's just what you think, so. All right, we see a lot of heterozygous. Homozygous dominant, let me just show you. R. If I cross these, all I'm going to get is a big R little r. So it can be homozygous dominant, but let's do the cross same way. Excuse me, sorry, but with a big R little r. So heterozygous, right? So when we do our cross, what do we have? Male up here. So we have RR, female down here. What do we have? Big R little r. What do we get in the first slot?
Big R, small r.
Second slot.
Big R, small r.
R little r.
Next slot.
Smaller, smaller.
Next slot.
Smaller, smaller again.
Small r's.
All right.
Shouldn't the third one
Something
be
feels weird here.
r's?
Something feels very weird here.
The
Shouldn't the third one be 2 small r's?
Yeah, that's what was weird. Yeah, I was like, something feels weird here. But yeah, so what's next?
Results.
Results. What do we put for results?
the genotype.
Genotype. What's our genotypes?
2 R, capital R, common R.
2
And
R.
2 recessive.
And
Homo-
2
homozygous.
homozygous recessive. Correct. So what's our phenotype now?
2 red rings.
2 red,
And 2 white.
2
2
white.
white. So perfect. So we know that this is the male. We know that this is the female. What's the answer to the question?
RR.
Which is?
Heterozygous.
She's a heterozygous female. Perfect. So let's deal with a bit of sickle cell anemia now. So inheritance of sickle cell anemia is autosomal recessive. It's the exact same as what you've been doing so far, except we just kind of write it a bit different. The two abnormal genes must be present in a child for that child to have sickle cell. So we can see two abnormal copies, we call them a sickle, but they're just affected. But you can just call them affected, but we call them a sickle. One abnormal copy, you're a carrier. 0 abnormal copies, you're unaffected. So a normal female mated with a normal male, and the result was a child with sickle cell. How is this possible using a Punnett square?
Mosquito.
So what do we start with?
The genotype.
What's our genotype of our parent? What's that? A normal male and a normal female. So when you're representing it, you do this, by the way. HB capital S, this is normal, and HB comma S is somebody with sickle cell, right? So I'm just giving you that kind of background information, yeah? And we can continue now, right? So what's the genotype of our parents? What's the genotype of the male?
They're both
HBSS is normal. HBS small s is a carrier. HB double small s's is a sickle cell. Is somebody with sickle cell. That's my answer.
About HBSS for the male, and then
SS.
Two
capital S's.
Yeah.
Yes. And then for the female, the HB capital S small s.
And the other one is what?
2 capital S's.
The first one is 2 capital S's. The next one is what?
Capital S small s.
And a common.
Capital S small s. All right. What else could the male's genotype be?
HB common S.
Yeah, it could also be HB capital S common S. If it was HB common S, he would have sickle cell, and he's a normal male. So remember, it's not X-linked, so it's not gamete. It's not, was it autosomal? And I can't remember, somatosomal or something like that. So it's not a genetic condition, right? It's not a X-linked condition. So let's deal with this one now, right? So we're dealing with our male now. So what does our male have? Our male has an HB capital S, and our male also has an HB capital S. So this is a common S, but what does our female have? An HB common S and an HB capital S and an HB common S. So let's put in our female here, and let's put in our male here. So when we cross them, what do we get?
For the first one, HB capital S, HB, and capital S.
Capital S, capital S. What do we get for the next one?
HB capital S, capital S.
What do we get for the next one?
HB capital S, common S.
Common S. What do we get for the last one?
HB capital S common S.
No,
Common
sir.
S. Capital S common S.
Oh, yes.
So results. What's the next thing I write?
Genotype.
Genotype. What's our genotypes?
2HB capital S capital S.
Next.
2. Wait, is it 2?
Yeah, this is a common. This is a common.
Yeah. 2HB capital S common S.
What's our phenotype?
2 normal and 2 normal carriers. Is that so?
2 carriers. So, do we see anyone affected here in this class?
No, sir.
No, sir.
So is this correct?
No, sir.
No, you need to have 2 carriers mating. So it has to be capital A, HB, capital S, common S times HB, capital S, common S. By the way, guys, you can just swat all of the results, by the way. When you mate 2 heterozygotes, what you get like half. You get 1 like this, you get 2 like this, and then you get 1 like this. When you mate, you get all recessive. When you mate a dominant and a recessive, you get all heterozygotes. Yeah, so stuff like that. You can just swap them. Like, no, because it said both of the parents were normal. So if one of the parents was HB2 common S's, then they would have sickle cell. So that can't be the case. Understood?
Yes.
Oh, send the stuff to the chat because it can benefit someone else. That question could benefit someone else, you know? So yeah, let's continue. So what we should have crossed is HB capital S common S with HB capital S common S. So I want you guys to do that cross yourself, right? So sexually linked characteristic inheritance. So inheritance of a sexually linked characteristic occurs only on the X chromosome, and only females may be unaffected carriers. So why? So when we look at our Y chromosome here, all that's on a Y chromosome is to say you'll be a boy. You'll be a boy. That's all that's on the Y chromosome. Well, the X chromosome carries a lot more information, right? So X versus XX, right? So let's continue. So a normal phenotypic female mated a normal phenotypic male, and the result was a child with hemophilia. How is this possible using a Punnett square? Remember, I think we did this question already, so I'm going to skip it.
Sir, could you go back, please? Okay, thank you.
So yeah, normal phenotypic female mated a normal phenotypic male, and the result was a child with hemophilia. Show how this is possible. I think we already did this one, but of course you have to have one X capital H Y. You have to have a carrier mating with an unaffected male. I'm not gonna do it again, but of course, every time that you're doing these things, follow all the steps, guys, please. So I think this is the last topic, actually. Are you guys ready for the last topic, or you want a break?
Continue.
Said a break.
Do you want a break or do you want just run into the last topic? Because I don't think it's actually that long.
One that breaks it.
All right, you guys get a minute break. No break. It's spoke too late. During the time, you can read the question if you don't want a break. I'm gonna get some water, actually. That's how I'm gonna use my break. All right, guys, ready again? If everybody's ready, say ready.
Okay.
All right. All right. So, can someone read, please?
During a medical examination of a 7-year-old girl, it is determined that she will be unable to produce adequate quantities of estrogen. Discuss how this condition may affect her future life.
All right. Ready. Hi, guys. How will this affect her?
She can develop fibroids.
She will have problem with during her menstrual cycle because like the uterus lining now get to develop.
Yeah. All right. All right, guys. So the answers are good, but they're kind of off base. The development of fibroids is not. I'm not so sure if it's specifically estrogen linked, but there's a chance that I'm pretty sure there's a chance that any woman can develop fibroids. I think it comes with age and not necessarily with lack of estrogen, although I think it's also associated with menopause. So that might be a good answer, but I wouldn't put fibroids. I wouldn't put the development of fibroids. I mean, she's 7, so what? There's a Brianna. Yeah, go ahead.
Well, that might it's talking about in her future life. Well, that might heighten the risk of her developing fibroids. That's why I said that.
Yeah, you're correct. I think you're correct, actually. I don't know. Let me look up the risk factors for fibroids in the meantime. But I think you're actually correct. But again, I wouldn't put that. There's easier answers to put. See, again, yeah, all of those look kind of like let's associate with menopause. Yeah, it's so good. No, I wouldn't put that answer though, but it's a bit much. Putting that is a bit much. I don't know the people that are marking your paper are gonna be like, this person is thinking. They're probably like, no, they didn't put what I wanted. So I wouldn't necessarily put that. About the uterine lining and menstruation. I wouldn't necessarily say you wouldn't be able to menstruate per se. But come on, guys, what?
I think
I wouldn't say you wouldn't be able to menstruate per se. But give me what's estrogen used for?
Sir, it could
Pardon me?
cause late in puberty, like if you
Perfect. Delay in puberty. What else? Come on, guys. What's associated with puberty in females?
About the menstruation thing. I think testosterone helps to bring to build up the lining. So if there's more testosterone, I think the lining would be more harder to shed.
I don't think testosterone plays any major role in females, really. All right, guys. No, all right. I'm just gonna say this. Testosterone is for males, estrogen is for females. Okay?
But there are some amounts of
when you think females,
testosterone
think
in
estrogen.
females.
If you're thinking testosterone, yeah, but yeah, when you think females, just think estrogen. Don't think testosterone. Just forget about testosterone. Because that just overcomplicates things, right? Because testosterone is by right a male hormone. But I mean, estrogen is derived from testosterone. So that's why you have some amounts of testosterone in females. Because estrogen is derived from testosterone. But what causes females to look like females is estrogen. So when we talk about females, we talk about estrogen. Just forget about testosterone for females at this level anyway. But I like where your head is at.
would it be difficult for her to get pregnant? I would say.
Yeah, sure. I'd put that.
Sir, it could cause a defect in the development of pituitary gland or the response.
Defect in the development of the pituitary gland?
No, sir, the response, like it wouldn't be fast.
No. Pituitary gland is a lot of stuff. Are you talking about? No, I wouldn't put that.
Okay.
I know what you're talking about, but I like where your head is at. I like where your head is at still. Because I know you're talking about the FSH and LH and the feedback from estrogen on the pituitary gland, right?
Yes, sir.
Yeah, but no, I wouldn't really put that because funny enough, it's not gonna slow down. If you don't have a lot of estrogen, these things work typically with like a negative feedback. So when you don't have a lot inhibiting it, it kind of like speeds up. But don't think about that. Forget about that. I'm just kind of telling you why that's kind of like not necessarily true. But don't get too into what I just said. That's not very important for you guys, right? Well, I don't think it's very important for you guys.
Sir,
irregular periods, correct.
And mood swings?
Irregular periods. What else? Guys, what else is estrogen used for? No, I wouldn't put mood swing.
Used to develop sex, right?
Used to develop sex. Sure. So I wouldn't say that specifically. What I would say is development of secondary sexual characteristics. So give me some secondary sexual characteristics.
Pubic hair, breasts.
Yeah, pubic hair. Yeah. Development of pubic hair. sparse. You can have maybe sparse pubic hair or absent. Where's do women grow hair?
And their arms,
Yeah. Guys, just look at yourselves. Armpits.
on your head also.
Yeah, but that doesn't necessarily have to do with estrogen. It's more underarm hair and pubic hair. So when you're thinking about this stuff, look at yourself. Think about yourself. Think about what you go through. What, monthly, I guess. So yeah, that's why it's really good to like deal with human-based stuff, because we're humans, you know. So development of pubic hair or arm hair, armpit hair is sparse or it might be absent. What else does estrogen help with? Maybe lack of breast development. But yeah, stuff such as those. The delay in puberty. Actually, yeah, that's correct. They would have a delay in puberty. Difficulty getting pregnant would be associated with the irregular periods. But yeah, stuff such as this. Yeah, you guys hit the nail on the head. You guys did perfectly.
Sarah, question.
Yeah.
So wouldn't estrogen like affect the lining of the uterus as well?
Why I don't want you to put that is because that it's kind of well, no, I think you guys actually have to know that. So I'll show you. I have a diagram of it, but estrogen is more for follicular development though. The Graafian follicle and all of that. What I'm saying is ringing a bell, right, guys?
Yes, sir.
Yeah. Estrogen is also associated with the uterine lining and the shedding during menses. But I'm pretty sure it's more progesterone that's responsible for that, and arterial constriction and sloughing off of the uterine wall and everything like that. I'm pretty sure it's more progesterone that's linked with that, right? I have a chart, so you guys can see. So the next thing, but yeah, estrogen is more for follicular development though, so. And also secondary stuff. So yeah. Which hormone, progesterone or estrogen, is present in greater quantities in birth control pills? Explain your answer. And this is also tied in with the with what you just asked, kind of. So what do you guys think?
Or estrogen, I think.
Estrogen.
Wait, no, progesterone.
Progesterone.
Sir.
Come on, guys, give more answers. Give more answers, guys. All right, just type it in the chat. Let's see, estrogen or progesterone. Let's have a debate, heated debate.
I'm not sure about this one.
That's fine if you're not sure. Just type something in the chat, whichever one you like more. Yeah, it's a fancier word, right? So I'm seeing a lot of estrogens. So no one says progesterone? So I guess progesterone is getting no love. That's fine. You can still know. Yeah, you'll just be the smartest grade 10 student. Probably progesterone. All right, so same progesterone, right? Let me explain my answer. you can cause childbirth. You wouldn't. Gabriel, unmute your mic and explain that.
Gabriel, sir.
Oh, Gabriel, sorry. Apologize.
Well, I mean, I think maybe estrogen can like cause childbirth in a way. I think I'm not totally sure, but if estrogen does do that, I think maybe need a different hormone to combat estrogen's effects on childbirth.
What's the hormone that signals childbirth is a fall off in progesterone.
Oh, okay.
So progesterone is high during
So-
pregnancy. Progesterone, it sounds like pregnancy. So it's high during pregnancy. And when that drops off, then you give birth to your child. And you have release of like you have other hormones that help out, which I'm not going to mention right now. But it's a fall off in progesterone, not, well, estrogen falls off as well, but the main one is progesterone. Progesterone is a hormone that maintains pregnancy, right? So let me explain why. My answer is, guys, what do you think my answer is?
Estrogen.
Estrogen.
No. My answer is progesterone. My answer is progesterone. Why? When you're pregnant, do you have a period?
No, sir.
No,
when you're
sir.
pregnant, when you're pregnant, can you get pregnant again?
No, sir.
No, sir.
So if I'm a woman, right? And I'm gonna take one bag of progesterone, what's gonna happen?
It's going to trick your body that you're pregnant.
Yeah, I'm just gonna trick my body that I'm pregnant pretty much. It's gonna make my body feel like I'm pregnant almost. So first, when you take birth control, you don't menstruate. That's one thing. I don't know how old you guys are. Yeah, let's not say that. Yeah, when you take birth control, you don't menstruate. That's one thing. So some people would like severe like dysmenorrhea, like painful periods, they just take birth control and they stop. They just cessate their menstruation, right? When you take birth control as well, you don't get pregnant because that doesn't what that has to do with because your body thinks you're pregnant, it's not gonna grow any more follicles, right? So you're not gonna grow any eggs to actually make you pregnant. So that's how that works. Cool.
Sir, birth control. So
yeah.
birth control stops ovulation.
Yeah, pretty much.
Okay. Sir.
That's actually what it does. Yeah, it pretty much stops ovulation, and it can potentially stop implantation. I guess could, but yeah. All right. So so let's write. So I said progesterone because prevents the development of Graphian follicle. Yeah, essentially, that's what I would put. It is possible to remove an ovum from one woman, fertilize it in a test tube, and implant the embryo in the uterus of another woman. So there's 3 problems which may be associated with this procedure.
The body may reject the egg.
The body may reject the egg. Post rejection.
It has a high chance of failing. I'm not sure.
Yeah, but why? Post rejection. I would maybe say insufficient hormonal hormonal level. Anything else? That's what I would say, because you need hormones to maintain your pregnancy. Anything else? Guys, anytime you're taking something from one person and sticking it up in somebody or implanting it in somebody, you can have potential scarring or things such as that, because you have to use an instrument, right? Scarring from instrumentation.
Okay, sir.
That's just a fact of life. Yeah, that's just something that can happen. So next, so some STIs are. Can you do it?
I was asking you to go back. And another question for number 1I.
1I, yeah.
So since the progesterone, when it's in birth control, and then it's going to like trick your body into thinking that you're pregnant. Like if you take a pregnancy test, would it be positive or no?
Sir, what's the word in front of follicle?
Graphian.
Sir?
Graafian. Graafian.
Okay.
Do you know how a pregnancy test works?
Honestly, I'm not sure.
Not really.
All right, so how a pregnancy test works, to put it simply, you have a hormone, right? The hormone is called HCG, human chorionic gonadotrophy. HCG. So HCG is present in the urine at two. So when this HCG is present in your urine, it just means that you have the formation of the chorion, the placenta. Placenta has to do with an embryo. So you clearly have an embryo being formed inside you, right? So if there's HCG in your blood, that means you have a baby. If there's HCG in your urine, that means you have a baby. So what do? That stick that you have, it actually has antibodies on there that respond to the HCG hormone. And so if the HCG hormone is present, it ends up changing colors, right? So that's why you pee on it. So of course, you're not gonna, most people don't suspect that they're pregnant literally a little bit after, or like 5 days after. It takes some time, right? For people to start suspecting that they're pregnant, right? Makes sense so far?
Yes, sir.
So you were breaking the
the hormone is human chorionic. Well, this is not something you know. I'm just telling you.
Sir, you are breaking up.
Because right? But the hormone is human chorionic. Am I still breaking up? Oh. You guys just told me that my internet is unstable. Am I still breaking up now? Hello?
We're able to hear you, sir.
All right, so the hormone is human chorionic gonadotrophin, right? That's the name of the hormone. That's being detected in a pregnancy test. So what does the chorion is just a fancy word for the placenta, right? I think. I'm pretty sure it is. It's detecting it's actually secreted by the placenta. So it's a hormone that's secreted by the placenta, HCG. It's responsible for the maintenance of pregnancy. And when that hormones becomes detectable in the blood after like a week, and in the urine after like 2 weeks post-fertilization, right? So so of course I said it becomes detectable in the urine, but nobody's just gonna be randomly doing a blood test, right? Just to check if they're pregnant, because I mean, how would you know, right? How would you know that you need to go and do the blood test to check that you're pregnant? It's people typically wait the 2 weeks and they realize, oh, I might be pregnant. I don't know. You're getting morning sickness, you're throwing up, emetic, stuff like that, right? Or you just feel general malaise. You just feel right? So like antibodies, the pregnancy test itself, that respond to the HCG that's in your urine, and that gives you a confirmatory color change if the HCG is present. If it's not, it stays the same, right? So I mean, that's how it's able to tell if you're pregnant or not, because if it's not in your urine, then you won't get any change, but if it is in your urine, then you will get a change. So you understand how the pregnancy test works now? Or kind of like the background information, okay, you don't need to understand how it works. I'm just telling you because you asked.
Right. Yes,
Oh,
sir.
yes, sir. Yes, sir.
All right, cool. So next, are caused by viruses and have been found to be without cure. Explain why viral infections are usually very difficult, if not impossible to cure.
Because they develop a resistance to the antibacterial medicine.
Can be antibacterials and it's viruses.
Antibiotics, sorry.
Antivirals, yeah. So develop a resistance, how? Yeah, they do develop a resistance, but how?
I guess mutation.
Yeah, mutation, simple. So viruses mutate commonly. Same thing with the COVID-19 and why it was so hard to come up with a definitive cure for it. You can't really cure viruses. And as such, it's hard to derive a definitive cure. All right, anything else? Anything else? Anybody knows what the latency period for HIV is?
Could you repeat that, please?
Anybody knows what the latency period for HIV?
No.
Good, sir.
Does anybody know what the latency period for HIV is?
No, sir.
It take 9 years or so.
Not so long.
6 years. 6 years. Sir.
Give me, it's a range. It's typically a range.
Somewhere between 9 and below 9.
Well, I've seen 3 to 7 years. I've seen 2 to 5. But yeah, something like that. So the viruses have a long latency period. So you can go a long time without even knowing that you're sick. So that's also why it's very difficult to treat. Because I mean, wait, give me a second. Long latency period. By latency period, I mean just a period that they go before actually starting to kill you. Viruses have a long latency period. Stuff such as those. All right, this part now, we can just move on. Apart from the nature of the AIDS virus, what other factors make the control of this disease very, very difficult?
So because it attacks the immune system, the body's defense system.
Apart from the nature of the AIDS virus. That's the nature of the AIDS virus. The AIDS is not a virus. Apart from the nature of the HIV virus. Sorry guys, I got this. This is a past paper question, so. Yeah. All right. When you
Because it
think
has
of,
no cure or vaccines.
sure, but not really. We don't really have vaccines. Well, I don't think we have vaccines. I think they're working. I think they're developing them. I don't know if they came up with them, but I don't think we have HIV vaccines. Spreading of the virus. Correct. That's correct, by the way.
So you could use a latency period because you don't know if you have it. So it spreads and
that's the nature of the HIV virus. So when I'm doing these questions, highly transmissible. Yeah, but again, that's nature. When I'm doing these questions, how I think about it biologically, think about it psychologically, I think about it socially. That's how I answer all of my questions. All of my questions when they're like control of diseases, this is how I think about them. Biologically, yeah.
Sure, so the answer for this question could be that it could be hard to control because people would stigmatize people who has it, so they will come out and say they have it.
Stigma toward infected individual. Perfect. So biological is nature. So give me more psychological and social. So why psychologically? Why would people with HIV not what would affect people with HIV coming out and doing whatever they need to do socially? So in society, what has to do with HIV? How does society treat people with HIV? Psychologically, how do people treat themselves when they have HIV?
Isolate themselves.
Right. So they don't really take care of themselves. I don't know. So I'm saying persons with HIV sometimes say they feel like all is lost, so they don't really take care of themselves or think that they're gonna die, so they don't really.
Yeah, that's the stigma. That's associated with the stigma. Psychologically. Anybody has anything else? Or you can give me something else.
Maybe they have the idea in there that they're eventually going to, but they rather not get treated.
That's kind of the same stigma. Move away from the stigma, guys. Kind of like broad, like think broadly. Psychologically. All right, psychologically, I don't know. You have to take a lot of pills. People don't like taking pills, right? So strict pill regimen, hard to follow. I don't know if you guys have grandparents and your grandparents take pills. Do they always take their pills? No. And they know that they're supposed to take their pills. They have the pills, they buy the pills, but sometimes you just don't take it. Sometimes you just don't remember to take it, or things such as that, or you want to go out or you want to do something else and you don't want to take it. So yeah. So psychologically, the strict pill regimen is hard to follow. So instead of pills, I would put antivirals. All right. So give me something else. Social kind of person, sexual, perfect. So how would we put that? Sexual behavior of the society. Polygamistic in Caribbean. They love when you put on the little in the Caribbean part, Yeah. So next. Guys, social education. Education is a social factor. People aren't educated about it. surrounding virus. So social education is always a social factor. Stigma is always a social factor. Literally, if you write out the stuff, biological, then social, then psychological, then social, you can answer any one of these questions, because this is how you answer all these questions. So I'm kind of giving you guys a little life hack, yeah? So, biological, just give me some biological. Although it said apart from the nature, just give me some biological stuff. That's just the nature of the virus. How is it spread? Stuff like that. The latency, stuff like that. Let me see what you guys come up with.
I think it attacks the immune system, and so it weakens its defense, basically.
Attacks immune system. It's easy to succumb to. Biological, what else? Anything else? Guys, just like to spit out answers. Come on. Again, the latency period. So a lot of people don't even know they have it. Another thing, the virus, again, affects the immune system. What cells? CD4 cells, I think it is. I don't remember. Yeah, probably CD4 cells. All right, latency period. What else? How it's spread. Injections, injection drug users, and sexual stuff like that. You just have to talk about how it's spread, the latency period, stuff like that. So yeah, that's the biological. So we can actually get into our last topic now. And we have like what, 15 minutes to cover that. So that should be more than enough time. So describe the menstrual cycle, roles of estrogen and progesterone, effects of pregnancy, and include the pituitary or gonads. I did not know you guys had to know about the pituitary before. But that's actually a lot. I think that's a lot. Go back.
Okay, thank you.
All right, cool. So let's talk about the menstrual cycle. So how many days does a menstrual cycle occur over?
28.
What days ovulation typically?
48 days.
What are the two phases of the menstrual cycle? Tell me if I'm asking you anything you don't know, by the way, because I'm just asking random stuff. You guys ever heard of the follicular and luteal phases?
Yes, sir.
Okay, good. So yeah, two phases, follicular phases and luteal phases. Which one is constant?
Follicular.
Luteal. Follicular stage just involves the growing of the Graafian follicle, and anything we grow has to be variable. Things don't just grow at a constant rate. Oh, when does menses begin? About what age?
Puberty.
About what age?
13.
Give me a number. 10?
After puberty.
10?
Yes, sir.
At 10?
After puberty.
No, that's a bit early.
13.
11.
11, 12, 13. 12, 13, 14. But yeah, perfect. All right, guys. So let's do this really quickly. So this is the perfect chart that you can ever see. This is the best chart you will ever see. This is a chart when I was studying for this last semester. This is a chart I use. So this is the best chart you'll see. So I don't know if you guys understand it, but I know when I was doing this stuff, when I was at CSEC, I didn't understand it because it looks complicated, right? It looks weird. So I'm going to explain it to you so you can understand. So again, two phases, follicular phase and luteal phase. So why is this called the follicular phase? Because you have the growth of the Graafian follicle occurring during this phase. Why is this called the luteal phase? Because you have the corpus luteum appearing within this phase. The hormones that are involved. So we're passing this forward. For the hormones that are involved, pituitary and ovarian hormones. So what are the pituitary hormones? FSH, LH. What are the ovarian hormones? Estrogen, progesterone. I wouldn't copy an ovarian hormone either, but whatever. So the endometrial cycle, this represents men. You guys need to know the various phases of the building up of the uterine wall. Proliferative, secretory, stuff like that. Let me know. Do you guys know? Can you hear me?
Sir, we don't know, but sir, if it is a part,
I'll just say. So you have a proliferative phase here. Proliferate just means to build up. So it's not anything too fancy, right? So proliferative, secretory. The good thing about the menstrual cycle, everything is what it says it is. That's the good thing. Secretory is just secreting stuff, like hormones and stuff like that. Specific stuff, not important. So let's move on. Development of the follicle. You start with the primordial follicle, you move on to the primary follicle, then the secondary, then the Graafian follicle, then the corpus luteum, then the corpus albicans. So let's follow the diagram, right? So we're starting over here. We're starting over here from the, well, we'd have a primordial follicle. We'd call this our primordial follicle. Maybe we'd call this the secondary follicle, the Graafian follicle. We'd call this our Graafian follicle. What's going on here? What's going on here? What's this process called?
Ovulation.
Ovulation. What day does this occur?
day 14.
The 14th
Approximately day 14. What's the first? What when it's left over? What is this? What is this called? The corpus luteum. And if we remember a phase name, what was this phase name called?
Luteal phase.
So why is it called the luteal phase? Because you have the corpus luteum. What does the word corpus luteum mean? The word corpus luteum, corpus just means body. Luteal means like yellow. So it's just a yellow body. So why is it called a yellow body? I'm not gonna say. But it produces progesterone. The purpose of the corpus luteum is to produce progesterone. So after ovulation occurs, then you start getting an increase in progesterone. Let's check it and see if that's correct. Ovulation has occurred right here. And then what do we notice? We're getting a spike in progesterone. So see, that's correct. So then after that, you continue being your corpus luteum, and then you become your corpus albicans. What does the word albus mean? Albus means white. Things that are white are almost like they're dead, right? So the corpus albicans, so the white body. So that's the end stage of the corpus luteum. So when pregnancy does not occur, it becomes the corpus albicans, right? So so let's deal with the pituitary hormones now, right? Let's deal with the pituitary hormones now. So first hormone that you have is FSH. What does FSH mean?
Follicle stimulating
hormone.
And when is that hormone high? During which phase? The follicular phase. Because that's the hormone that the follicles need to grow. Okay? So that's why it's high during the follicular phase. So the next hormone that we have is estrogen, right? why do we have that hormone suddenly being produced? That hormone is being produced by the dominant follicle or the Graafian follicle. Sorry, give me a second. So yeah, this was our dominant follicle. So this was the follicle that would be producing estrogen. All around this ovary, you actually have follicles in different stages, by the way. So it's just one of them actually becomes mature and starts to secrete estrogen and stuff, secrete estrogen and actually goes on to give you the egg. Just one. Okay. So when you're in the womb, you have like what? Like a million ova. You have like a million ova. When you're born, you have like what? I think 500,000. By the time you reach puberty, you have 40,000, I think. I think you have about 40,000. And only one out of those is actually ovulated. Only one of those actually develops and gets out. But at a time, you have many trying to develop at a time, but only one gets out. So that's important to know. So estrogen is secreted by what secretes estrogen? Which I just wrote, the Graafian follicle. All right, LH comes from what does LH come from? LH is a pituitary hormone as well. FSH comes from what?
The pituitary gland.
FSH and LH come from the pituitary gland. LH, what does LH mean?
Luteinizing
hormone.
What does luteinizing mean? Luteinizing is just like the signaling of ovulation. So when this LH starts to get high, then that's when you start to release this. That's when you release this ovum. So LH spikes at around day 14. Why? Because that's when your ovulation is occurring. So again, progesterone associated with the corpus. The corpus luteum. And what do we see with these uterine walls when the progesterone is increasing? What do we notice about them?
It's built up.
They're thick, right? They're thick. Yeah. So that's pretty much it. That's the menstrual cycle. There's nothing else really. And when pregnancy does not occur, you form the corpus albicans, you have a drop off of progesterone, and all of that is shed. That's all menstruation is. So the functions of estrogen and progesterone. So estrogen, they're both steroid hormones. Estrogen produced by the Graafian follicle. What produces estrogen?
Graafian follicle.
What produces progesterone?
Corpus luteum.
Skip the rest. Yeah. Yeah, what's the function of estrogen?
For the growth of the uterine lining.
Yeah, I don't really want you to put that.
And it stops the secretion of FSH.
Remember, estrogen starts with an E, ovulation. O estrogen, ovulation. Yeah? So we're keeping it simple, right? We're keeping it simple. Estrogen, ovulation. So estrogen is associated with ovulation, right? What's the function of progesterone? They're all there, guys. I'm asking you stuff where the answer is on the slide. I'm not gonna ask you stuff that I'm not telling you.
Breaking up.
What's the function of progesterone?
Yeah, it's the uterus for pregnancy.
What's the function of? Okay, yeah, we can continue. So yeah, good job, guys. there's not much else to say on that topic. That's pretty much it. It's not so very complicated. What I would like is for someone to explain it to me. So, who can I ask? Who's been answering my questions? Joel, are you there?
Yes, sir.
Can you please explain to me the whole process from the whole process of the menstrual cycle? And you have diagrams.
All right. So first of all, the pituitary gland, secretes FSH, which is an hormone which stimulates the growth of the Goffreyan follicle.
Stop for a second. Stop. What does the menstrual cycle start with?
A soar in estrogen.
Starts with menstruation.
It does. I thought it ended with menstruation.
No, that's the first. See, cycle starts with menses.
All right, so the uterus lining from the previous cycle sheds, disintegrate, and is lost to the vagina. After which, as I said before, the FSH secreted by the pituitary gland will be secreted, and it will cause the growth of the Goffreyan follicle. After which, the Goffreyan, so you hear me?
I'm hearing you, but
it has a low bandwidth. It's saying low bandwidth on my end, that's why. Anyways.
Oh no, it's fine.
The pituitary gland now will secrete Allele, luteinizing hormone, which will...
Wait a second. You didn't talk about estrogen.
Estrogen, did I?
Yeah.
Oh, all right. The Goffreyan follicle secretes estrogen with source during the second part of the follicular phase. After which,
what's the function of estrogen?
I know one function out of my head, and it's to stimulate the growth of uterus lining. So that's another function.
Okay. All right. Development of secondary sexual characteristics. This is estrogen. Estrogen allows for development of the uterine lining. I don't have a problem with that. Ovulation it allows for the continuation of the maturation of the Goffreyan follicle. Again, secondary sexual characteristics is actually the main function of estrogen, right? So continue.
Okay, sir.
So what's the next hormone?
Yeah. After the estrogen, now the pituitary gland secretes luteinizing hormone, which next water.
Which?
So I'm not here.
It secretes LH,
Hearing, sir, or are you guys not hearing?
Carry the hair. I carry the hair. Double. Oh, I think it was on my end because my broadcast stopped talking again. Right.
So I was saying that the after the estrogen source, and you said it's used for secondary sexual characteristics, right?
Yeah, I did.
That's the main function at that point. I'm going to move on and say the pituitary gland secretes luteinizing hormones, which stops or slows down the production of FSH And this luteinizing hormones makes the ovulation take place. After ovulation takes place, there's a slight surge in the in estrogen.
Yeah, forget estrogen at that point. Talk about progesterone.
All right, yeah. So the Graafian follicle forms into the corpus luteum, which is otherwise known as the yellow body. And this yellow body secretes now progesterone. Have stopped being secreted. The progesterone now builds up and maintain the structure of the uterus lining, preparing it for infertile implantation and fertilization occurs. In the event that fertilization doesn't occur, it turns into a white body, which stops secreting progesterone. And
no, you can't just say white body. You have to actually say the name.
I forgot the name.
Corpus albicans. Corpus albicans means white body, just like how corpus luteum means yellow body. So you have to say corpus albicans. Yeah.
All right. Yes, sir. And that,
you know, like the word you watch Harry Potter, like Albus Dumbledore.
Oh yes.
Anybody watches Harry Potter, you know, like he has white beard.
Yes, sir.
Like white body, white beard.
Yes, sir. Corpus albion. Yes, sir. And that stops progesterone from secreting. This ceasing of progesterone causes the uterus lining to disintegrate and menstruation occurs. This only happens if the egg isn't fertilized, and both the egg and the line leave the body through the vagina.
Yeah. Talk about the follicular development. Actually, someone else. Yeah, good job. Really good job. Can someone else tell me about the follicular development? What are the stages of the follicle? Hello, anyone there? Can you guys hear me?
I don't remember. I think I remember one.
Primordial, primary, secondary, Graafian.
Oh yes, sir, Graafian.
Primordial just means ancient, so like it's the first one. It's like the first one. Primary, secondary. Graafian just means the main one that's developing, and then you have the formation of the corpus luteum, and then the corpus albicans. So can someone just repeat what I just said?
Can I write these things in words, please?
All right, so it goes primordial follicle. Get your primary. So I just wrote the word twice for some strange reason. Primary follicle, then you have your secondary follicle, right? Then you have your Graafian follicle. Then you have your corpus luteum. Then you have your corpus albicans. Yes. Yeah, Joe, go ahead.
Sure, question. In the event that fertilization actually occurs and during the pregnancy, possession. Yes, sir. You hear me?
Yeah, I'm hearing you. Yeah. But good question. Yeah, continue.
All right, I'm saying that during the pregnancy, after fertilization occurs, during pregnancy, is progesterone secreted straight throughout the pregnancy?
You're breaking up, kind of. Can you type the question in the chat?
Yes, sir.
All right. Well, I know what you're asking, and I'm just gonna answer. So, all right, you know that you have the corpus luteum going to the corpus albicans if you're not pregnant, right? So when you're pregnant, the corpus luteum becomes a corpus luteum of pregnancy. So literally, it's just what it is. Like the name tells you everything. So the corpus luteum becomes a corpus luteum of pregnancy because what's the function of progesterone? To maintain the uterine lining. So you wouldn't want a lady with a pregnant lady with her her fetus just suddenly aborting, because menstruation with a fetus is abortion. Is spontaneous abortion. So you wouldn't just want that to happen, right? So what you would want instead is the maintenance of the uterine lining, and that's the function of progesterone. So you need the progesterone to continue. So what happens, the corpus luteum of pregnancy continues secreting progesterone, and the placenta also, the placenta, you guys know about the placenta, I'm sure. You hear about that stuff, like rich people eating their placentas and stuff like that. Yeah, you hear about that stuff, like the rich people on like the entertainment network and stuff like that. They do that stuff. So the placenta actually secretes progesterone as well, and it also secretes the HCG, which they both help to maintain pregnancy. So once you're pregnant, you keep secreting the progesterone. If you're not pregnant, the progesterone falls off. That's why you see the progesterone dropping down, by the way, because that progesterone dropping down is the signal that, okay, we're gonna menstruate. If the progesterone stays high, we don't menstruate. Yeah? And if the progesterone stays high, then the cycle does not restart. Yeah. So I hope you understand. Does that answer your question, by the way? All right, great. Excellent. You can lower, yeah.
Wait, so
all right, so let's talk about the pituitary. Yeah.
The corpus luteum dies when the female becomes pregnant, since the placenta is going to produce the progesterone more?
Yeah, essentially. After a while, if you get pregnant, you don't need the corpus luteum because the placenta is going to produce the progesterone, but you do need the progesterone. But you don't need to know that. I'm just telling you because, again, you asked. So, yeah. By the way, guys, it's really important that you watch over this recording, like really important, and just kind of like cement these stuff. But yeah, I think you guys really should rewatch this recording. So let's do this now, the pituitary gonadal axis. So ignore this part. So from the pituitary gland, you get LH and FSH. What does LH mean?
Luteinizing hormone.
Luteinizing hormone. What does FSH mean?
Follicular stimulating hormone.
What does FSH do? What does follicular stimulating
Is that
hormone
the hormone that tells the follicle to start grow or something?
To start to grow. It's the growth. It makes a follicle. Yeah. Again, guys, all of this stuff, it's like this stuff is kind of like great. All of this female genital stuff is great because all this stuff are explained by the name. So luteinizing hormone, that what does that do? Triggers ovulation. Perfect. So from the gonads are just the testes and pay no attention to the male side. Pay no attention to the male side, just look at the female side. So the gonads for the female, what's the female gonad? The ovary, guys. The ovary is a female gonad. Estradiol is just estrogen. Estradiol is just estrogen. And progesterone. So all this is saying is that when the pituitary secretes LH and FSH, it signals the female ovaries to produce estrogen, and eventually progesterone. Right? Yeah. So that's all you guys. I think that's all you guys have to know. Do you guys have to know about negative feedback like that? No, right? Oh.
No, sir. Well, not for the topic.
All right, great. So let's move on then. All right. So birth control pills, synthetic progesterone, so they maintain quote unquote pregnancy. And if you're pregnant, you can't have a follicle being stimulated. The follicle is not going to grow. If the follicle is not going to grow, then what does that mean? You can't get pregnant really. This is just some forms of birth control. So for this one, I'm not going to go through it specifically. I just want you guys to read it or take a picture of it or write it down or something. This is something that could really come on your exam. So just know them, know this stuff, right? I have some pictures over to the side as well. All right, let's continue. All right, let's look at STIs now. Pardon me?
Thank you, sir.
All right, yeah, definitely. So let's look at STIs now. So what does STI mean?
Sexually transmitted infection.
What does STD mean? What
Sexually
will happen if you take birth control while pregnant? Why would you? Birth control gives you acne and those stuff, you know. Why would you? And
like if you didn't know, I get to that.
oh, and you were pregnant? Probably nothing. Probably nothing. You'd probably still have the same. You'd probably still have the child.
So you're not gonna affect you.
No, you'd probably just get acne and the other things that are associated with taking birth control, like increase risk of clots. Yeah, birth control has a lot of side effects. Nobody's just gonna be popping birth control just because. It has a lot of side effects. But yeah. And after a while you should know. So I don't think you just maintain a birth control and you know you're just pregnant just like that. You'd realize eventually that you're pregnant. So STI, so condom, that's a barrier method of control and protection against STIs. So these are some STIs examples: HIV, gonorrhea, chlamydia. All right, yeah, let's move on. I'm not gonna answer that. So these are some examples. So I'm just breaking down the word HIV. What does HIV mean?
Human immunodeficiency.
Human immunodeficiency virus.
So this is just breaking down that whole thing. So human, it attacks humans. Immunodeficiency attacks and kills immune cells. So that's why you have a deficiency of immune cells. Virus is a retrovirus. It's just a virus. Yeah. So that retrovirus is just a type of virus, by the way. AIDS, acquired immune deficiency syndrome, acquired of infection. So because you get HIV, then you get AIDS. Immune attacks the immune system. Deficiency, low immune cell count, so it's killing the immune cells. Syndrome is just a list of disease characteristics. Syndrome is just something you have, right? So again, AIDS is the disease, HIV is the virus that causes it. So know that. So again, that's why there's no AIDS virus. Yeah. So again, HIV virus transmitted via bodily fluid from an infected individual. So you can have transmission via blood or serum transfusion, needle sharing, unprotected vaginal or anal intercourse with an HIV affected individual. Syndrome is just like, all right, let me give you an example of syndrome. Syndrome is just a list of disease processes that you just have. Sorry, not disease processes, just list of symptoms that you have, and then they call it a syndrome, right? So if you have certain disease characteristics, then they say, okay, you have this syndrome. Do I know any other syndromes? All right, so let me give you an example of another syndrome. So there's like Marfan syndrome, which is like something that affects your call it your bones. Call it affects, well, it doesn't really affect your bones. It affects like your cartilage and stuff like that. So these people, what do you find when you have Marfan syndrome? You I think you have abnormal long limbs. You're prone to developing like aneurysms and stuff like that. So it's just it's just a list of characteristics that you observe in a person with a certain disease. That's all I would define a syndrome as, right? So again, unprotected vaginal or anal intercourse with an HIV affected individual. So that's where you're having sex with a woman or a man. Exposure to ulceration of the skin. Yeah, you can get HIV that way, right? So HIV transmission influenced by, as I said, how I break it down, biological, psychological, social. So biological, long variable latency period, and it's dormant. So again, people a lot of times don't even know that they have it. Viruses, a feature of viruses is that when they replicate, they mutate. So it's really hard to treat viruses, really hard, right? So it's hard to vaccinate against viruses. Viruses in general, that's just the nature of the virus, of viruses in general. Psychological. So there's a stigma attached to those with the virus, and people avoid getting tested. And even when they get tested and know they have it, they avoid getting treated. And some people, even though they know they have it, they just go around infecting people just because. Crazy. Social. Unwillingness to use barrier protection methods such as a condom. I don't know, some people okay, nothing really to say about that. Lack of education surrounding anti-HIV prophylaxis medication. So if you actually have if you have sex with somebody who has HIV, you can have, I think it's up to 36 hours, you can have post-infection prophylaxis treatment, which actually reduces your chances of acquiring HIV. But a lot of people, again, don't know this stuff. So if you're a high-risk individual and you know about this stuff, you can take it. But if you don't know about this stuff, then there's no urgency for you to take it. If you guys understand what I'm saying. You guys understand what I'm saying? You can just type in the chat or just unmute your mic and let me know.
Understand.
Anybody doesn't understand so far? Okay. Okay. I guess that's the end. So yeah, thank you guys for coming. I went about 23 minutes over the time, so thank you guys for staying and still being attentive. When the recording is off, I'll take any questions that you guys have, or if you guys have any questions now from the session. But if you guys have just generalized questions for biology, I can help you with that. If there's a topic you don't understand and I can just try to brush through it quickly, I could help you with that. But again, thank you guys for coming. What's that? Maybe like 17 of you that are probably left by now. Yeah, I hope it really helps. So you guys can let me know if you think it helped or not. And guys, remember you still have to oh yeah. Guys, remember you still have to study at the end of the day, but this should be a good start up for you in your studies, or because I kind of try to include all that you. All right, cool. I'll just do that quickly. I kind of try to include all that you guys might need to know. Kingdoms, what about it? I don't know. What is it? Kingdom, phylum, class, order, family, genus, species. All right, menstrual cycle, cool. what's this? King Philip something, I don't remember, but you guys can come up with your own mnemonics. So if I was come up with a
I think
mnemonic,
it's their King Philip comes over for good sex or good soup.
King Philip comes over for good sex and or soup, whatever. Guys, any way you can remember it, just use that to remember it. So if soup is what you like, use soup. If you like the other thing, use that. So yeah. Anything else about there's not much to really do about that. Kingdom is at the top, species is at the bottom.
Repeat how to like remember it.
Use mnemonics. So when I'm doing my stuff, I just make up mnemonics. So when you look at K, you look at the first letter K, P, C, O, F, G, S, you can literally say anything. You can say give me something that starts with K. I'm gonna say kill. Keep ponds clean or frogs get sick. That's a great mnemonic. Keep ponds clean or frogs get sick. I was gonna say some foolishness, but yeah, that's an excellent mnemonic. Keep ponds clean or frogs get sick. Keep ponds clean or frogs get sick. See, so yeah, you know this stuff. You know how it's going down. Kingdom would of course have the most organisms, and it gets more specific as you go down. Stuff like that. Yeah. So I don't remember. I say, guys, this is not what I do in my day to day. So I wouldn't really remember like the specifics of this stuff, because I think you're right. One of these is like a common letter, or I italicize something and stuff like that. Like that's not stuff I bother myself with day to day. But for the menstrual cycle, that's stuff I bother myself with. So I can help you with that again. So going over it again quickly. You have your FSH. FSH from the pituitary signals the growth of your follicles. Growth of your follicles, you get a dominant follicle, the Graafian follicle, which starts to produce estrogen. It's not me that's dealing with that. It's not me that's dealing with the recording. So estrogen, which allows for the growth, the secondary sexual characteristics, and it does have some impact on the uterine lining, but I don't like that answer. I don't like that as the main answer. LH, luteinizing hormone, released by the pituitary, signals the start of the luteal phase and or signals of ovulation. Next thing, we're going from the follicular phase into the luteal phase. Luteal phase, we have the corpus luteum being produced. Corpus luteum starts to secrete progesterone. So let's check our uterine lining now. So first we start with menstruation, then we start to proliferate, build up the uterine wall until we reach the secretory phase now. We start with primordial, then we go to a primary, then we go to a secondary, then we go to our Graafian follicle. We get ovulation coming from that, and then we have our corpus luteum, corpus albicans, corpus luteum. This is the initial corpus luteum. Then we have our corpus luteum again, corpus albicans. That's all menstruation is, guys. So yeah, follicular phase is variable, luteal phase is constant. I'm sure any girl can tell you that when your period comes, is kind of unpredictable, because some days you might think that you're pregnant because you haven't seen your period in like a week. But yeah, but you haven't seen it in like a week. But you know, it's still on track, because just remember the follicular phase is variable. So yeah, anything else? Any other questions? Anybody say anything in the chat? No. So you guys good?
Sir, remember we were going over the sickle cell thing. I was wondering if it was only females that could be a carrier.
Only females can be a carrier. Yeah, only females can be a carrier. Sorry, no, not for sickle cell. Sickle cell isn't genetic, isn't sexual. So yeah, yeah, males or female can be a carrier for sickle cell. So only
Sir,
yeah?
what's your card?
Yeah, of course. So anything else?
So please post the PowerPoint in the group, sir.
Yeah, I'll think about it. You guys have a recording, so why you need the phone?
It's a recording going
I'm
on.
kidding. I'll send the well, I'll think about it. But yeah, thanks for coming out, guys. That's the end, like the official end. If you guys have any other things you want to ask, just ask them in the group and I'll do my best to answer. So yeah. Thanks for coming. I got to Yui. Can you end the recording and I'll just talk about that quickly? How do I end the recording? Because I want you guys to get the recording now.